Dear colleagues from European regulatory medicine agencies, dear representative of healthcare professionals, dear representative of patients, I am so delighted to welcome all of you to the Real World Evidence Conference organized by the French National Agency under the framework of the French Presidency of the Council of the European Union. The use of the real-world data is a growing field in the development and monitoring of health products thanks to the increased abilities in big data collection based on upgraded computer and digital capacities. The convergence of new treatments, treatment modalities, diagnostics, medical device, wearables, sensors, and connected health is generating enormous amounts of data. More and more routine healthcare data are captured in electronic format. Principal sources of real-world data include electronic health records, claims data, and data from registries. The use of the real-world data to generate evidence of regulatory decision-making is already a reality now, and there is increasing interest in using big data and advanced analytics as a complementary source of evidence. Big data as the source of real-world evidence is a current and huge challenge and a real opportunity for every stakeholders, and this is part of the European Medicines Agency's network strategy to 2025 for protecting public health at a time of so rapid changes. There is an increasing emphasis on post-approval activities using the real-world data. Clinical trials remain the foundational method of establishing the safety and the efficacy of medicines during the pre-authorization phase. However, they do not fully reflect the world, resulting in gaps between regulatory dossier and subsequent clinical evidence, which are needed by downstream stakeholders, including HTAs, but also payers, and surely, ultimately, clinicians and patients, which digital techniques have the potential to address. This current World Evidence Conference today you are attending, is a unique and will be primarily focused on the post-approval activities. Indeed, throughout the COVID-19 pandemic, a huge amount of real-world data have been generated and analyzed in order to provide subsequent evidence on safety, but also on efficacy of vaccines that were developed with an unprecedented effort to reduce duration and to accelerate their provision to the population, meaning to hundreds of millions of people. This conference will be a unique opportunity during the first session to present feedbacks in the field of pharmacovigilance, but also on pharmaco-epidemiology, as well as the input in the decision-making process and communication to the population. Beyond COVID-19 pandemic experience, there are obviously ongoing initiatives among European member states that allow further development of analytical capabilities, including integration of data and analytical methodologies. This second session of this conference will be also an opportunity to share such experiences, questioning the interoperability of database and platform construction, the quality of the collected data and their representativeness, the data process analytics, and the digital tools in successful or several less conclusive attempts reflecting the operational reality. Then, a third session will be dedicated to the existing networks and the global initiatives to coordinate the works made in the field of real-world evidence in order to evolve data-driven regulation, facilitating the development, authorization, and supervision of medicinal products. This is, for example, the HMA-EMA Joint Big Data Steering Group that aims to advise heads of medicine agencies, but also the European Medicine Agencies Management Board. This is also the DARWIN initiative and the Health Data Hub developments. We could not talk about real-world evidence without also presenting the interest of the real-world data as a complement to the clinical trials for medicine in the pre-authorization phase. For example, in the field of rare disease and pediatrics, where a comparative randomized trial may be very, very difficult to perform. Real-world data may also support evidence on the natural history of the disease being treated and on population beyond those represented in clinical trials. And we will talk in the fourth session about the registries and viewing real-world data as these complementary tools to clinical trial aiming to benefit patients through better decisions based on more comprehensive evidence. I wanted this time to be really a bottom-up conference by stressing on the importance of the initiative across Europe member states. There are the main bricks. The European member states are really the main bricks for any European construction and networking. Sharing on successes or less conclusive attempts, most importantly, debating, questioning all together is ought to be the best way to go forward on such tentacular topic. Experiences will be shared from France, from Norway, Denmark, Italy, Spain, Germany, Finland, and the Netherlands, and we will generate, I hope, and I am sure, very stimulating discussions. A roundtable will finalize the two-day conference that will focus on the two major thematics. The skills that are really needed to be developed in the regulatory agencies, and I am particularly sensitive to this, the place of the patient in the system. And we will be pleased to hear at this roundtable the place and the voice of the patients by the means of one of their representatives. Before opening the conference, I would have a thought dedicated to the Ukrainian people part of the European population, who is today suffering and encountering such a very dramatic situation. Let's hope all together that the European community emerge stronger from this disaster. Now, this is the time to open the conference, and I would warmly thank all the speakers for having accepted to present their involvements and works in the field of the real-world data. I wish you two very fruitful days, and I would give now the floor to Laurence Baril and Pierre Desmolis, who will moderate the first session dedicated to the COVID-19 vaccines. Thanks a lot.
So should I start? Okay, so welcome everyone, and very happy to introduce you to that conference on the real-world evidence for 2 days. Laurence Baril and myself are responsible for the first part, and I would like to inform you on some housekeeping rules. This conference will be registered, and there will be opportunity for you to ask questions. The questions could be asked by the internet. So depending on the channel you're on, you can ask the questions in French or in English, doesn't make any difference. And we have two colleagues in charge of selecting the questions, Samuel Cromlin and Youssef Chang, both from the French agency. And we will have the first set of questions and answers after the two first presentations. So this being said, it is my pleasure to introduce Mehdi Benkabil, who is the Deputy Director of the Pharmacovigilance Surveillance Directorate at the French agency. And Mehdi has a PhD and is a specialist in real-world data in the collection of large sets of data, organizing the data and making them available and ready for statistical interpretation. So he's very experienced in the topic. And he will now present the reinforced pharmacovigilance of COVID-19 vaccines as it was operated in France over the last month during the pandemics. Thank you, Mehdi. The floor is yours. Thanks a lot.
Thank you, Pierre. Good morning. So ladies and gentlemen, good morning to you all. I'm pleased to present you a reinforced pharmacovigilance of COVID-19 vaccines surveillance in France. So just give you an overview of this surveillance, which has been put in place in France. So as part of the national vaccination campaign against the COVID-19, ANSM has set up a specific system for reinforced surveillance of the adverse effect of COVID-19 vaccines. So the objectives of this surveillance is to carry out a continuous assessment of the COVID-19 vaccine safety in order to be able to ensure their safety or to quickly take the necessary measures in case of any signal. The second objective consists to communicate in real time on the result of this surveillance in order to provide transparent and exhaustive information on the result of this surveillance. So this monitoring is based on two arms. Two important arms, sorry. So the first one is related to the pharmacovigilance. So it's based on the analysis of adverse effects which are reported by the healthcare professionals and patients to the national and European system. And the second part is based on the pharmaco-epidemiology studies. So I'm going to focus on the first part, which is the pharmacovigilance system. And the second part will be more detailed by my colleague Mahmoud Zurek later on. So first of all, I would like to just describe you and explain what's the pharmacovigilance. So the pharmacovigilance has the objective to monitor, evaluate, prevent, and manage the risk of adverse effects, which result from the use of drugs, of medicines. So this could be, could occur under normal condition of use, as well as in other situations, such as overdose, abuse, medication errors. In France, what we have, the pharmacovigilance is active at regional level through 31 regional pharmacovigilance centers, at national level through ANSM. The main tool in pharmacovigilance is the spontaneous adverse events, which are reported by the healthcare professionals and patients and the patient associations. So now, what consists of this reinforced pharmacovigilance of vaccines? So the challenge was to collect potential signals in real time related to any unexpected adverse effects and any particular interest of these adverse events, such as Kawasaki diseases, Guillain-Barré syndromes. So this reinforced pharmacovigilance consists of seven aspects. The first one concerns the reporting. So what have been done is to enhance the reporting web portal in order to collect specific and relevant information related to the vaccination. Also, we were proactive. We had proactive communication on how to report adverse events. The second part concerned the detection. So on this part, it has been performed detection and cross-analysis of different data sources in order to raise any particular signal. So potential signals are raised by the regional pharmacovigilance centers, and also another source was the scientific literature, where monitoring were performed at the national level. The third aspect of the reinforced pharmacovigilance of vaccines against COVID-19 consists of the analysis of all adverse reports within pharmacovigilance surveys. And we have set up in total 5 surveys. So it was organized as following. So we had 4 expert pairs in total, 1 pair per vaccine. We had a specific survey on the pregnancy, which involved 2 experts, and 1 expert pair for the coordination of all this survey. We had also put in place 2 additional experts for a Comirnaty pharmacovigilance survey, and in total, we had 13 experts. We also involved experts in specific area, type of pathology and physiopathology, such as the cardiology, neurology, and so on. So all these experts were involved in order to make an expertise on the cases reported to the pharmacovigilance system. Also, what has been put in place is stronger collegiality. So a specific monitoring system was set up, which involved experts from ANSM and regional pharmacovigilance center. So in case of a safety signal is validated, measure adapted to the nature of the risk and the level of the risk is put in place. And these measures will aim to prevent or reduce probability of occurrence of the risk in the vaccinated persons. Also, our stakeholders were involved within the ANSM committees. So, another aspect of this reinforced pharmacovigilance of vaccine against COVID-19 is the reactive and agile system, depending on the knowledge of the risk profile of vaccine. So we were able to change our manner to work. So we changed from weekly to quarterly pharmacovigilance survey reports. We introduced a new kind of reports. So by producing a monthly focus for specific population and subject of interest, such as pediatric teens, booster doses. Also, we adjusted the frequency of the monitoring committees, going from weekly then to the bi-monthly. Also, we had stronger transparency by going from the weekly to bi-monthly production to the end of at the end of the monitoring committee. So we published the pharmacovigilance survey report, focuses, summary sheet, integrating the key figures of the pharmacovigilance data and significant result. Also, we produced operational information for healthcare and patient in order to explain how to proceed following a signal. And finally, we support and answer to citizens' questions and media. And finally, the last aspect of this reinforced pharmacovigilance is more coordination between the pharmacovigilance and pharmaco-epidemiology, the 2 arms of this surveillance. So specific studies, pharmacovigilance, pharmaco-epidemiology studies has been conducted in the event of emergence of a pharmacovigilance signal. So now I'm going to focus on some aspect of this reinforced pharmacovigilance surveillance. So concerning the reporting, so we improved the web portal of the adverse event reporting. So the form has been improved by collecting new information concerning the vaccine, how the vaccine has been administered, the history of the COVID vaccination. Also, we produced information sheet to user in order to explain how to report adverse events by focusing on serious or unexpected adverse events. The second aspect of this reinforced is providing adverse event information sheet intended for the healthcare professional and patients, of course. So it's a brief sheet which explains what kind of adverse events could occur and the frequency and how you manage these adverse events. Also, as I said, we produced a guideline in order to explain how to report the adverse events. So it's a clear guideline which explains each step of this reporting. And finally, concerning the process of the reporting, we put in place some enhancement. So these figures illustrate the process of the reporting, starting from the patient, association patient, and healthcare professional reported. It could be performed by 2 channels, through the web portal, directly to the regional center by phone or email. So the improvement has been put in place by making an interconnection between the web portal and the database, the National Pharmacovigilance database. And also what has been implemented in this pharmacovigilance database is an artificial intelligence module which gives priority and case pre-coding. But at the end, the medical analysis is performed by the expert from the regional center. Also, another aspect and final aspect is an example of the summary sheet here, which gives key figures of the surveillance. This is an example of the survey of the pharmacovigilance survey reports. And finally, we produced some information concerning how to manage in case of adverse events. And these are 2 examples concerning the flu-like syndrome, and the other one concern the vaccine-induced immune thrombocytopenia. And concerning the reinforced coordination between pharmaco-epidemiology and pharmacovigilance. So these studies are carried out by the EPIPHAR, which will be more explained by my colleague Mahmoud Générique. And here are some examples of studies. Just to give you one example on my side, it concerns the occurrence of myocarditis and pericarditis in people aged between 12 and 15. And this study has been carried out after raising a signal from the pharmacovigilance. And this will be more explained by my colleague Aurélie Grandvillemain and then Mahmoud Zoureik. Concerning the European coordination, so in case of a potential signal detected at the national level, this is systematically reported to the EMA level, so that a scientific assessment will take in place in coordination with all European countries. And at the end of this assessment, if the signal is confirmed, an update is performed on the summary of product characteristics. Also could be issued an additional risk minimization measure adapted to the nature of the risk. Finally comes to the conclusion. So as of March 3rd, 2022, we can say that we collected a huge number of adverse events. It's 145,000 adverse events reported concerning 140 million doses injected in France. And what has been set up is 43 monitoring committee meetings. We produced 67 pharmacovigilance survey reports, 17 focus, and 3 specific points for all vaccines. So these key figures demonstrate that the national reinforced surveillance system was able to support the vaccination campaign and confirm the safety profile of all vaccines in view of current data. We also can say that this national system is completely complementary to the surveillance which took place at the European and global level. And finally, we could say that the national surveillance system maintains an agility to respond to the future challenges. Thank you for your attention.
Thank you very much, Dr. Mehdi Benkebil, and good morning to all. Now we will move to the second session. And I have the great pleasure to introduce Dr. Aurélie Grandvillemain. She's a pharmacist. She has a long-lasting experience with leading the Regional Center for Pharmacovigilance in Burgundy. She's also the vice president of the organization of the 31 regional centers for pharmacovigilance in France. She has had a leading role in the several reporting and focus for the COVID-19 vaccine. So thank you, Aurélie, for your presentation. And after our presentation, there will be some discussion. So please introduce your question in the chat already. Thank you.
Thank you, and good morning to all. And thank you for giving us the opportunity to present the French Regional Pharmacovigilance Network, especially this year through COVID-19 vaccine safety monitoring. So could I have the slides, please? Thank you. Let's start from the beginning with a few steps of history of pharmacovigilance in France. It starts in 1973 when the 6 first hospital pharmacovigilance centers were created following the WHO monitoring recommendation for drug safety. And pharmacologists in a university hospital endorsed this new mission. 3 years later, pharmacovigilance entered the French law by the first decree that described pharmacovigilance activities. In 1978 was created the Association of Pharmacovigilance Centers that I have the honor to represent today. In 1982, The French pharmacovigilance system was more precisely described in another decree in law. In 1984, the obligation for health professionals to declare to report adverse drug reaction was written in law. The French pharmacovigilance database was created in 1985 by the RPVCs, and the 31st Regional Pharmacovigilance Center was created in 1986, and by now we are 31 RPVC across France. And the last step I would like to underline is in 2011 with direct reporting by patients that is permitted by the law. Next. After history, let's have a look on the main characteristic of this pharmacovigilance expert territorial network. First, we are locally implanted in pharmacology departments in university hospitals. That permits a close link between pharmacovigilance centers and health professionals, and also with patients. Next. We are medical structures, managed by pharmacologists, which are medical doctors and pharmacists, and we provide a phone permanence 5 days a week for health professionals, but also for patients. And next. We are independent structures with exclusive public funding. Next. And as I said before, we are a network with constant exchanges between us, between RPVCs, and also with a close relationship with ANSM. And next. Briefly, what are our main missions and activities? First, we provide a clinical and personalized expertise for drug-related disease through a help for diagnosis, drug risk assessment, and risk prevention. Next. We are also directly involved in health monitoring by identifying drug safety signals at regional level, but also at national level, as explained by Mr. Benkebil. Next. We do expertises in pharmacovigilance, drug risk prevention, and proper use of drugs for different kinds of structures, healthcare institutions, regional health agencies, and also at national level with the ANSM. And next. We also do research, information, and training for students, healthcare professionals, and also general public. Next. Let's go deep in pharmacovigilance in practice. All starts with a disease that could be drug-induced, and this situation are notified to RPVCs. But often the question is which drug may be involved in this kind of disease. And so this leads the notificators, that could be medical doctors, pharmacists, patients, nurse, and so on, to contact the RPVC to have help for diagnosis and also advice for care and support for management of this drug-induced disease. So the pharmacologists in the RPVC do the job, medical and pharmacological expertise, and give the conclusion directly to the notificator by phone and or by mail, and this mail is included in the medical chart of the patient. And next. If the adverse reaction is suspected, so the case is recorded in the French pharmacovigilance database and sent electronically to ANSM, and the French database is implement the European database, Academia-regulator, which belongs to the European Medicine Agency. Next. If a potential safety signal is identified by or suspected by the RPVC, the case is immediately and directly sent to ANSM through a special way that is called significant case. Maybe it's not a good translation, but this case are then discussed into committees or taken into account directly if it's very urgent. And these significant cases represent around 10% of the total cases the APVC received. And then if the signal is taken, it's validated at national level, then it's sent to Europe to be discussed at a European level. Next. And the real difficulty is to answer this question. Is it an adverse drug reaction? So to do causality assessment, we work on 4 elements. The first one is chronological evaluation, which consists of reconstitution of the story. I mean, when was the drug administered, date of beginning, date of stop, at what dose, when began the first symptoms, also the more precisely possible, and which drug were taken at the same time, what is the outcome. So you can see on the slide a small figure that consists of all drugs administered with date of starts and stop, and the evolution of the adverse reaction that permits to analyze more precisely the involved drugs. Next. The second step is a semiological evaluation, which consists to collect all the symptoms that could be drug-related and to determine if these symptoms are related to a specific disease. I mean, for example, a headache could be a headache, but it also could be the signs of hypertension, or could be the first symptoms of cerebrovascular accident. So, do these symptoms have been explored? Or are other etiologies being excluded? Indeed, adverse drug reaction is often a diagnosis of exclusion. And at the same time, we do a pharmacological evaluation to find a potential physiopathological link between the disease and the drug. And next. And finally, a literature analysis is done to know if this adverse drug reaction have been already described, and also to find information for management, for care and support of this adverse drug reaction, to give advice to the notificator. Next. In view of all these points, let's see how COVID-19 vaccines monitoring is a real pharmacovigilance challenge. First, there was at the beginning no insight with this new vaccine technology and new target, I mean S protein of this new virus, SARS-CoV-2. And next, the exposition is also extraordinary, with a very large number of people exposed in a very short period of time. This was never seen before, even with flu vaccines campaign. And next. And it should also take into account the specific perception of vaccines compared to more classical drugs. I mean, vaccines are drugs that are administered to healthy persons, and the individual benefits is delayed. And the benefits is very particular because it's not to catch a disease, so it's not tangible for many people. But the risk could be immediate when you get vaccinated. And another particularity is the mechanism of action of vaccine through immune system that makes sometimes adverse reaction more difficult to diagnose and to understand by some people. Next. For pharmacovigilance structures all over the world, and also in France, of course, the challenge resides on the caseload volume and high proportions of patients' notification. As you can see in this figure, you can see the number of cases recorded in the French pharmacovigilance database since 2000. And you can see the first 10 years, the increase was slow, And there is a first increase in 2017 with the implementation of a vigilance web portal that Mehdi spoke about a few minutes ago, and also with the levothyroxine crisis in this year. 2 years later, it go back to normal life, if I can speak like that. And as you can see, in 2021, the huge increase of number of cases recorded in the French pharmacovigilance, nearly 100, I have problem with 160,000 cases recorded in the French pharmacovigilance, with a high proportion of COVID vaccines, of course. And you can see in orange the proportion of patients' notification. That is nearly 40% in 2021 compared to 13% the 2 years before. Next. The increase is not only seen in the French pharmacovigilance database, but also in the number of questions and advice asked from patients and practitioners to RPVC, because as we spoke before, these new drugs with the potentially unknown adverse reaction and the media and political pressure for people to get vaccinated generate lots of questions. And as you can see in the figure, we have, compared to the 2 previous years, nearly 2 times the questions were multiplied by 2 and spontaneous notification nearly by 5. And next. The challenge consists also in medical expertise of the case received. I mean, we receive more adverse events than usual rather than adverse effect. I mean, some people report as adverse events related to COVID vaccines, medical events that appear in usual life, and the causality assessment is sometimes very difficult in such situation. And another challenge was the low informativity of initial notifications. And we all know the importance of documenting serious and or unexpected adverse drug reaction to have a better signal detection and also better signal validation. I mean, headache is not the same safety signal than hypertension or cerebrovascular accident. And next. Let me show you an example of what kind of data we received in RPVC. So this is a declaration form of a case of myocarditis received through the web portal. So next, you can see we have the regulatory data with the notificators information, patient information, also suspected drug, and the adverse reaction. But next. As you can see, there are lots of missing information. I mean, relevant history was not fulfilled, the clinical, biological, and imaging data, and also the outcome. So next, here you can see the science forms of the same case recorded in the French Pharmacovigilance database after documentation by the RPVC and pharmacologist expertise. And as you can see, we can obtain the relevant history, concomitant drug, the clinical, biological, imaging data, the outcome that permit to better describe the adverse reaction itself, but also its severity and management of this adverse reaction. And next. To be able to face this pharmacovigilance challenge and to be able to do our main job, that is fast signal detection and also good quality reports for signal validation, the RPVCs have put in place prioritization of activities to respond to the context, with a priority given to response to question and advice from health professionals and also patients, and that takes a lot of time. And the priority given to documentate and record in the database the serious and or unexpected adverse reaction related to vaccines. And also don't forget the adverse reaction, serious and or unexpected of other drugs, because they still be used, of course, at the same time of vaccination. Next. We should also be flexible in vaccine safety monitoring. I mean, the people exposed to the vaccine evolves over time. At the beginning, we have seniors in institutions in January, and then teenagers in June, children in December, and the data collected, the way to collect the data, the adverse reaction reported, the questions are very different among these different populations. And we also should have to document specific points of attention, like in March, the Vaxzevria adverse reaction reported by healthcare professionals, and many emerging safety signals that need to be documented in priority. Next. In the survey context, we put in place harmonization procedures for coding in the database, that evolves day after day. We also worked to make the significant cases where to report directly the signals to ANSM more relevant day after day. We also make lots of information exchange within the network between RPVCs and also a very close collaboration with ANSM. And for RPV experts, the additional workload was very, very significant with the daily cases review, and keep in mind the high number of cases we received every day, and weekly reports, weekly committees, and many more. And 20 minutes is not enough to make the list complete. And next. And we should notice that we have support from ANSM, first with specific exceptional financing that permits staff reinforcement. That unfortunately was not as high as the charge of work, but it helps. And we also have technical support with a new database put in place in April that permits interconnection with the web portal, and also artificial intelligence which was put in place for patient cases, but was not so helpful, to be honest, at this time. To illustrate the wave that goes from spontaneous reporting to signal identifications, let's speak about the example of myocarditis. This adverse reaction was added to SMPC in Europe of RNA vaccines in July. Next. By then, between July and September 2021, the experts saw a differential increase in case notified between Spikevax and Comirnaty. The number of cases for Spikevax was multiplied by 11 in 2 months, compared for Comirnaty, which it was multiplied by only, if I could say it like that, 4. Next. The second finding was in September, where the experts of Spikevax found another representation of patients below 30 years of age for myocarditis case, with a notification rate which was 0.5 for 100,000 injections, compared for people below 30 years of age, and the notification rates was 3 for 100,000 injections. Next. So in early October, the experts do a specific analysis which compare both vaccines, Spikevax and Comirnaty, and for the different class of age. And they found a significant results for Spikevax for men after second dose of vaccine and for people below 30 years of age. And the notification rate was 19.2 for Spikevax compared to 3.4 for Comirnaty. Next. At the same time, the Scandinavian countries brought the use of Spikevax to people below 30 years of age. Next. 1 month later, a pharmaco-epidemiological study conducted in France by EPIPHAR confirms this pharmacovigilance signal. And this study will be presented to you soon after. Next. All this data lead the French Haute Autorité de Santé to modify the vaccine recommendation, and by now Spikevax is not recommended anymore for people under 30 years of age. Next. To conclude, I would like to remind you 2 key points. The first one is the success of the vaccination campaign is based on safety monitoring and the safety use of vaccine, transparency of the information, and the effectiveness. And next, the second point is to have an optimal drug safety monitoring. You need to have a rapid feedback from the field and also good quality of case reports to have better signal identification first, and then to permit validation of the signals identified. Next. To be able to reach the main objective of pharmacovigilance, that is fast signal detection and good quality of reports, the French regional pharmacovigilance network provide a real-time pharmacological and medical expertise of each adverse drug reports that RPVC received. And next. This activity is done by an experienced territorial network of 31 centers managed by pharmacologists. Next. The key lies on reactivity and flexibility to adapt the monitoring to the context. And next. One strength of the RPVC network is its regional and national approach that provides a close link with patients and healthcare professionals, but also a close relationship with health authorities, ANSM in particular. Next. We could resume the action of pharmacovigilance with this figure that is going from patient to signal and then go back from signal to patient and clinician advising. Patients are really our priority. And thank you for your attention.
Dr. Grandvillemain, and thank you both for your presentation showing the importance of a strong collaboration and also, the need for this collegiality and agility to face challenge. So we have time for a few questions from the audience. The first one I may ask to both of you is, do you think that this integrative organization you have set up for COVID-19 vaccine would be sustainable in the future? And could you express potentially your need to strengthen those organization?
I don't know, maybe if I start. What we need is hand, arms, and people to help us for working on all these cases we received. As you can see, the activity was very huge last year, and the beginning of 2022 is the same. We have lots of questions and notifications, and I think if we want a pharmacovigilance system effective, independent from industries, we need to have more pharmacologists in regional pharmacovigilance centers to do the real medical job.
Okay, thank you, really. Mehdi, any thoughts?
We don't hear you, Mehdi.
You can hear me? Yeah.
Okay.
Okay, you can hear me?
Yeah.
Yeah, perfect. So it's more or less in place at lower level. I mean, today, yeah, the number of reporting was 5 times as normal, as already showed. And we have such organization in the pharmacovigilance within the committees which are in place today, but it's not at the same frequency, of course, but it's more or less similar what has been put in place for the COVID-19 vaccine surveillance. So this is something which is, I mean, more or less in place, but it's not at the same speed. It's we can say that it is 5 times less speed than the what has been put in place for the COVID-19 vaccine.
But the collaboration with ANSM exists for other drugs since many years, and we work also together for other surveys. But as you say, Mehdi, it's not the same volume of work.
Yes, it was very intense, even based on a strong pre-existing organization, I can imagine. A second maybe question is related to the fact that you have to face also all the aspect of lay communication with healthcare professional, but also with the citizen, with the public. Could you share about this experience and thought also for the future about this important part, you know, of the system?
Okay. Maybe I can start. Yeah. Thanks, Laurence. So, I mean, the communication, we had a high level of communication, of course, and related to this communication, we have, of course, a lot of questions. So more you communicate, more you have questions. So it is a very good opportunity for us to have this level of transparency, and I mean that gives a good feeling from the healthcare professionals and citizens. So I mean that it's still in place. I mean this kind of communication is still in place for drugs, but it's not at the same level. But I mean, we are still on the way to improve what is in place today. Aurélie, maybe you want to add?
No, I think transparency and scientific discussion explain to people with simple words how it works, what we know, what we don't know at this time. People understand. We had lots of patients that call us in RPVC, and when you explain to them where we are, what we know, what we don't know, they understand. So I think it's very important to be honest and to explain what we know and what we don't know, I think.
Okay, thank you very much. We have to take care of the timing. So I propose that Pierre will introduce the next speaker, and we move from France to Norway now. Thank you.
Thanks, Laurence. Thanks. The next speaker speaks remotely from Norway. And thanks a lot, Pernille Haag. Forgive me for the pronunciation if it's wrong. Pernille is the alternate member at the Pharmacovigilance Risk Assessment Committee centralized for the European Medicines Agency in Amsterdam. So she meets the colleagues on a regular basis every month. And she has been working for the pharmacovigilance system for over 20 years, and over the last almost 2 years, she has been responsible for the coordination of the pharmacovigilance activities in Norway for the COVID vaccines. So Pernille, the floor is yours, and thanks a lot once more for having accepted participating in that teleconference. Thanks a lot.
Thank you so much for the kind introduction, and to the ANSM for the invitation and the opportunities to share the experience of COVID vaccine surveillance from the perspective of a country with a quite small population. I will focus on the organization of surveillance, the methods used, illustrated by some examples, and share some experience on communication and transparency. May I have the slides, please? And the second one. Thank you. I will start with a short overview of the setup of the Norwegian corona vaccination program. Like in many European countries, the vaccination campaign is organized and rolled out by the National Institute of Public Health. The 5 main areas of responsibility of the program is listed to the left in this slide. One of the main areas, as you can see, is surveillance. Surveillance include both surveillance of vaccine efficacy, which is the responsibility of the Norwegian Institute of Public Health, and also safety surveillance. Safety surveillance of the vaccine is mainly the responsibility of our agency, the Norwegian Medicines Agency, NOMA. In the planning phase, the safety surveillance plan was divided into 3 main areas, which you can see on the bottom right. The first 2 were planning for handling large volumes of adverse drug reactions, ADRs, and signal detection and signal evaluation, which I will explain in more detail later. And we also plan for communication of safety information. Our communication colleagues have been an integrated part of both planning and pharmacovigilance activities during this whole period and participate in all meetings. Even though the responsibilities are split, we have worked closely with our colleagues at the Norwegian Institute of Public Health throughout the pandemic, both on the pharmacovigilance side and on the communication side. Next slide, please. Nationally, the surveillance of the COVID vaccine have rested on 2 pillars. The first one is the spontaneous reporting of ADRs. As has been explained, the reporting of suspected ADRs from clinical practice from both healthcare professionals and vaccinees. This method is of course well known to us and is known as a quite rapid, simple and easy signal generating method. By the way, we receive almost exclusively electronic reports to our spontaneous reporting system. In addition, in Norway, we have quite a few national health registries covering the entire population. As the second pillar in surveillance, we plan for using these data to support pharmacovigilance. The most important national health registries are listed on the slide. The Norwegian ADR database was, luckily, just prior to the pandemic, turned into a national health registry as well. As we are in a pandemic, we also have a temporary emergency preparedness register for COVID-19, where all these data sets listed and quite a few more are pooled and analysis can be performed across different registries. This is made possible by the fact that every individual in Norway have a unique personal identifier or a social security number, which makes the individual identifiable across registries. A lot of these registries are updated overnight, which makes it possible to use these data both in almost real-time surveillance and in more long-term surveillance of the ADRs. In practice, combination of these two methods has been shown to be very useful. As a potential signal has shown up in the spontaneous reporting system, we have been in the position to fetch data from the registries that either support the signal or refute it by showing that the reporting is in line with the background frequency. Next slide, please. So a little bit about the organization used to handle the large volume of ADRs. The basic thought was that you in crisis should rely on systems you know to work and have experience with. So the organization diagram shown is the standard organization. As shown, we have 4 regional pharmacovigilance centers in Norway, located at 4 large regional hospitals. In addition, as you can see on the top of the chart, we have 1 national center for vaccine ADRs, located at the Norwegian Institute of Public Health. These 5 centers handle healthcare professional reports only. The Norwegian Medicines Agency is responsible for the procedures used for ADR handling at these centers, and NOMA is also the host of the ADR database. Please click. When speeding up this system to handle the large volumes of ADRs received for the COVID vaccines, experienced ADR handling personnel from the regional centers were reallocated to handle healthcare professional reports for the National Center for Vaccine ADRs, as shown in this with these pink arrows. At the peak, about 20 additional staff from the regional center participated in the handling of vaccine ADRs. Please click again. In addition, the Norwegian Medicines Agency have during the pandemic vaccination hired extra staff, about 20 persons, both full and part-time, to handle patient reports. About 75% of the reports we have received to date are patient reports. Internally at the agency, a number of staff has also been reallocated to COVID pharmacovigilance for shorter or longer periods. In spite of this extra staff, we still have a backlog of non-serious ADRs. Next slide, please. In addition to the human resources side, we also quickly saw the need to organize the daily work in a way that would meet the challenges of the large volume of data coming in. During this period, the data and signal management has gone almost hand in hand as all processes have been sped up. This slide is very busy, but tries to summarize the main activities put in place. First, on the top left, there is the triage. Triage of incoming ADRs is done on a daily basis. In the triage, the fatal, serious and unexpected ADRs are sorted out and prioritized for case handling. We also prioritize all reports regarding children. So in the top middle, we have had short morning meetings. In the start, these meetings were held daily, and at the moment, it's twice a week. In these meetings, we discuss fatal cases, cases which are currently of special interest or in our focus, and prioritize our daily work. On the top right, there is an illustration of an internal group which we call the signal detectives. They are responsible for the daily signal detection, which has included following all new data coming in, detection of striking cases, and looking into our adverse events of special interest. They have prioritized cases for further internal discussion and cases that require follow-up. Then to the lower left, there is our weekly internal meeting where pharmacovigilance staff, physicians, vaccine specialists, and PRAC members have had a broad discussion of interesting cases and assessed and prioritized these for potential escalation. The weekly ADR publication has been discussed, and this meeting has also served as a preparation for the weekly meeting the following day with the Institute of Public Health. This common meeting, which is another Teams meeting, which is illustrated in the lower middle, has served as a forum for information exchange between NIPH and NOMA. We have discussed the need for registry analysis based on potential signals detected in the spontaneous reports, and these analyses are performed by the NIPH. As mentioned, communication colleagues have also participated in these meetings, and we have discussed the need for public communication of particular issues and the weekly ADR publication and news item. And last but not least, we have of course also used international data sources such as the Eurovigilance Data Analysis System, WHO's Vigilize, which we use extensively in our signal detection, and of course other international sources of ADR's information. As a country with a small population, international data is very important to us. Through this system, we have decided which issues to follow nationally, which signals should be raised at a European level to PRAC, and communication at the national level. Next slide, please. I hope I've managed to give you an overview of the organization of pharmacovigilance in Norway. So now to the more practical side and examples. Prior to the start of vaccination in Norway, we started the communication process. We warned or advised the public that the vaccine had short-lived but often unpleasant ADRs, as shown here to the right, and that some would have to stay home from work for a day or two. We prepared the public that ADRs were more frequent than we are used to for other vaccines. We also informed the public that for the adenovirus vector vaccines, the ADRs seemed to be most pronounced after the first dose, while for the mRNA vaccines, it was the second dose. And that younger adults were more likely to experience ADR than the elderly. We also knew that the vaccination in Norway would start with the elderly and most fragile in nursing homes, and that normally an average of 400 elderly die every week in Norwegian nursing homes. So we prepared the public that deaths would appear shortly after vaccination, likely without any causal relation to the vaccine. Next slide, please. When we started vaccination, our expectations were met in the way that deaths in the elderly, fragile patient were reported to us. Within a few weeks, more than 20 deaths were reported, and within 2 months, 100 deaths in patients in nursing homes. These were patients with underlying disease, and they died from their underlying disease. But in some of these cases, geriatricians found that common ADRs like fever and vomiting may have contributed to the fatal outcome of their underlying disease. It was and still is an expectation from the public in Norway that the authorities are transparent and communicate ADR data, which we did. And also our Minister of Health, shown in this picture, conveyed this message to the public. This was handled quite calmly in the Norwegian press and did not create a lot of attention nationally. But as I will come back to, it created a lot of attention or havoc internationally. Apart from this picture of new ADR, the new ADRs we saw was not so alarming, and is summed up at the right in this slide. This changed quite rapidly in the beginning of March 2021. Next slide, please. This is when we first heard of cases of patients with coagulation disorders, bleeding and thrombosis. Due to the limited time, I will not go into all the details in this timeline, just highlight that the Norwegian Institute of Public Health paused the use of Vaxzevria in Norway as a precautionary measure upon news from Denmark about a fatal case. This was to buy us some time to gather information and clarify the situation. Within the next 2 days, we received information about several patients in Norway with thrombosis, low platelets, and bleeding. Within a few hectic days, we received a lot of details from the reporting physicians and informed the European network via a rapid alert, as we found these cases to be a potential signal. At the same time, the NIPH also started analyzing our national health registries to look for potential further cases and background frequency of cerebral sinus venous thrombosis in this age group, etc. Next slide, please. Days later, a laboratory at another university hospital specializing in this showed that all the patients had autoantibodies to platelet factor 4, which led to aggregation of platelets. They said this looked like heparin-induced thrombocytopenia, but was not heparin-induced. At the same time, corresponding findings were made in Germany. At this point, precautions were added to the EU SPC of Vaxzevria just 10 days after the first notice of a problem. I will not go into all the details of the further EU process for updating the SPC, just say that after 1 month, by the 7th of April, what we currently know as TTS or VIT was included in the SPC. We know of 7 clear cases of this syndrome in Norway, and 4 of these patients unfortunately died. A lot of member states paused their vaccination for a short or longer period. In Norway, the use of Vaxzevria was permanently stopped. The vaccination program has since, and up until now, only offered mRNA vaccines, used in mix and match combination. This is a sad story, which we had hoped to avoid. However, I think it's an important illustration of why pharmacovigilance is vital and why we do this work. Next slide, please. I will dwell just for a little while on why WIT became a strong signal in Norway in the important first phase. Some of the elements we believe contributed were, firstly, as a country with a small population, some specialized hospital functions are centralized. This means that all the Norwegian cases were admitted or transferred to the same department at Oslo University Hospital. They got the first 3 patients within 1 to 2 days. This similar, unexpected and very atypical picture of thrombosis, low platelets and bleeding became evident to them, and the doctors sought for common factors. The only common factor they could find was the vaccination with Vaxzevria about 2 weeks earlier. Secondly, Vaxzevria was given mainly to healthcare workers in Norway, which means that the patients had been basically healthy and with few risk factors for disease prior to this event. Thirdly, we have physicians who have an active reporting culture and also took direct contact with the authorities. And finally, there is a very close contact and collaboration between the clinical departments and research units in Norwegian hospitals. The 2 doctors pictured here and their teams dropped everything in their hands and contributed 24/7 to identify part of the pathology quickly and kept informing the authorities. Next slide, please. As I mentioned, we also use registry data to support the VIT signal. Based on the well-documented ADRs and the potential signal detected, we could quickly specify the need for analysis on both cerebral sinus venous thrombosis and bleeding. These analyses strengthened the signal and helped with details on frequency, etc., which formed a better basis for communication. This form of collaboration is new to us, but has proven very useful. But the Emergency Preparedness Register for COVID-19 makes this easier to do in almost real time. In a normal situation, it would currently take longer to get access to data and the required authorizations to do coupling of different health registries. In addition to the health registries, we have also used another link between pharmacovigilance and pharmaco-epidemiology during this pandemic by following up potential ADRs in cohort studies. We have several established cohorts in Norway which have been sent questionnaires regularly for a long time, also during the pandemic. Next slide, please. Menstrual disorders is an example of a potential ADR where we have used questionnaires to the cohorts to gather further information. From early summer 2021, we received an increasing number of reports of menstrual disturbances and vaginal bleeding disturbances from women. At the moment, we have thousands of reports of a variety of menstrual or bleeding disorders from women of all ages. The main reason for using cohorts and not health registries for looking into this issue is that we understood from the patient reporting these disorders that they had not necessarily sought medical attention, so the events are not captured in the registries. So the NIPH included questions on menstruation and bleeding in the questionnaires for the existing cohorts. The data analysis is still ongoing, but the preliminary results show a high background frequency of menstrual disorders and an increased rate of heavy menstrual bleeding in 18 to 30-year-old women, but further data are being analyzed. There's an ongoing signal procedure in the EU for heavy menstrual bleeding and amenorrhea. This Norwegian study is included in the analysis, but all other data sources will be included in the assessment, which is still not finalized. Next slide, please. So as a last point, I will share a little bit about communication of ADRs and transparency in Norway. Since 14th of January 2021, about 2 weeks into the vaccination campaign, we have been publishing a summary of reported ADRs, the ADRs we have received. Well, the report was weekly until December and bi-weekly since then. In the Nordic countries, the public trust in the authorities is quite high. This is, at least in part, believed to be due to transparency and openness about uncertainties in the knowledge base for the vaccination program, and as I mentioned in the beginning, our promised openness about the ADRs reported. We have also been open about the backlog of non-serious cases due to the large volume, and we ensure the public that all fatal and serious cases and ADRs in children are prioritized for processing, and we are up to date on those. The weekly report is also translated into English. I will shortly come back to why, and is available on our website. Next slide, please. We are used to this openness and transparency in Norway, and luckily, in most cases, Norwegian press provide balanced information, and we believe that the public generally understand the information given in a nuanced way. But we learned the hard way in January last year that vaccines are very political, and media in other parts of the world operates very differently. So while the previously mentioned fatal cases in nursing homes did not really hit the headlines in Norway, it did in the international press. And a lot of the important nuances in the Norwegian communication was lost in translation by Google Translate. More than 500 international media had headlines like seen in this picture, and what we thought was a plain national communication was used in the international political play. There is obviously a lot to learn from this, and one of the things we learned was to translate the weekly report to English so that all nuances were kept. Next slide, please. And just to sum up the numbers of reported ADRs in Norway per 1st of March, we have received almost 58,000 corona vaccine ADR reports. For comparison, in a normal year, we receive about 5,500 for all drugs. Almost by chance, we planned for a tenfold increase in ADRs, which turned out to be very close to reality. By 1st of March, 51% of the reports are handled and submitted to Pharmacovigilance. We have 255 reports with a fatal outcome. And of the handled cases, 19% are serious, but that number will be reduced when the backlog is handled. And 55% of the are from patients or vaccinees, and 80% for women. Of the total volume included the backlog, 77% is from patients or vaccinees. Next slide, please. To sum up a few of our key learning points. Close and frequent monitoring of incoming spontaneous report with focus on striking cases is of great value, and it focuses the surveillance and the need for public information. The integration of pharmacovigilance and pharmaco-epidemiology gives a better knowledge base in a stressed system. It is helpful and important when signals arise during ongoing mass vaccination, and we think it has been key to patient safety that it has been possible to perform these analyses so fast. And last but not least, communication and transparency is paramount for trust, but very demanding and resource intensive. Next slide, please. So thank you for your attention. I'm happy to take any questions.
So thanks a lot, Pernille. It was very clear, and we could see where the differences are. And maybe one of the important points of your topic was the sake of transparency. In the Nordic countries, I wouldn't say that these exercises are not performed in southern countries, but the consequences could be a bit different for cultural and organizational reasons. But we may come back to that. The discussion. The first question coming from the network is because Norway and the other Nordic countries have a strong experience with registries and cohort studies. Do you see differences in the establishment of the pharmaco-epidemiological approach for COVID-19 vaccines as compared to previous vaccines?
Yeah, I think obviously we have been able to do this much faster than we usually do due to the access to this emergency preparedness registry for COVID-19, and I think that is really important and a key learning that these data must be available on a very short notice, and we can't wait for applications and delayed access to data. So I think that as is part. I heard your questions a bit. Well, it was a bit difficult to hear, but I hope I answered your question.
No, but I take from your answer that because we had something as an emergency, we should go fast to vaccine people. The kinetics of the reactions should be completely different as compared to other vaccines, where we have plenty of time to adapt. And this is something we had to face in every country. The next question is, was the communication different as compared to other vaccines introduced in the population? And is the model of transparency with the integration of communication department very early in the system something that was invented for COVID or that applied to other pharmacovigilance issues?
Well, I think we always work very closely with our communication colleagues, but I think we early saw the need for this. So it has been more consciously done, I think, in this route. And I think when it comes to the openness and transparency and the frequent communication, I think obviously we do not communicate like for other vaccines every week, but like for the HPV, we have published reports every 6 months because it has been a lot of attention around that in Norway. So I think we have experience, but not doing it this often, and with all this attention.
Thanks, Pernille. And once more to share our experience in building on the cultural differences possibly. How could you deal with some external opinion that many adverse reactions are not reported, not notified to authorities, and as a consequence are unknown? This is an opinion which is that opinion present in Norway or not? And if yes, how could you deal with that?
I'm sorry, could you repeat the first part? I lost the sound for a bit.
Sorry. So there could be an opinion that some adverse events are not reported because of the defect of the system. And some could say that what we work on is a partial recollection of the adverse events, because many of them not being reported are unknown, which could be a mistrust in the signals we are dealing with. So is that opinion present in Norway? If yes, how do you deal with that? If not, do you believe that this is a cultural difference with other countries?
Well, it's difficult to say, but I think from the volume we got, we understand that quite a lot is reported, but obviously not everything. But we also tried to convince, well, via the communication in the public, that you don't need to report a headache and fever and all the known ADRs. You need to report what is different. And I think, well, they report known ADRs as well. But I think at least the healthcare professionals, I think they're quite aware of this, that we mostly focus on what is unknown, so we can detect the new ADRs. So there might be cultural differences, but I think healthcare professionals in Norway have a good reporting culture. So I think the communication between us and the healthcare professionals is good, I think.
Both directions, indeed. So my next question is to the PRAC representative. As a PRAC alternate member, what is your experience on how the PRAC could cope with the problem over this period, and how beneficial was for you the sharing of the data and the conclusions, the discussions that took place during the PRAC sessions?
Well, I think the PRAC has shown that it can adapt to different circumstances or a crisis, as during, well, mostly last spring, there were an awful lot of extra meeting and ad hoc expert groups and everything organized to handle the situation. And I think they've shown that they can, well, step up in a crisis. And I think, well, with even though not an EU member, Norway is a part of this European collaboration around the marketing of drugs and everything. And I think we couldn't do this alone. I think we are dependent on the international or European collaboration, and it's also that we need this to, well, we cannot update the SPC in Norway and everything. We do that in a European way. So I think it's, well, it's really important for us being a part of this international network.
Thanks a lot for that answer. I would take that as a take-home message, and it wasn't part of the introduction made by Christelle, Ratignier, Carbonneil. Europe is a network. We must work with that network for the best of our tasks, and this is an incredible opportunity for sharing, trusting each other, and having a much more powerful system. Thanks a lot. I believe this is the end of this early morning session. Then we will have a 10-minute coffee break, so we should come back within 10 minutes from now. So I leave you the calculation, and it will be for the next session more focused on the pharmaco-epidemiology. Thanks a lot to the speakers. Thanks a lot to the audience. Thanks a lot to our colleagues responsible for the chat, and thanks a lot to you, Laurence. Bye.
Thank you, Pierre.
Welcome again to the real data conference. I'm delighted to introduce you my assistant, Claire Ferrard and Evelyn Pierron, who are going to collect your questions. So please don't hesitate to send your questions. So now we're going to start the second part of this first session concerning the safety of COVID-19 vaccines. So I'm pleased to introduce you Mahmoud Zoureik, who's going to present the presentation concerning the pharmaco-epidemiology studies concerning the myocardist and pericardist after COVID-19 vaccines, and also the efficacy of anti-COVID-19 vaccines. So I give you just a brief presentation of Mahmoud Zoureik, who is a researcher and research director at the French National Institute of Health and Medical Research. He joined the French National Agency for the Safety of Medicines and Health Product in 2012 as head of epidemiology of Health Products Division. Since the end of 2018, he is director of EPIPHAR. EPIPHAR, it's an epidemiology scientific interest group. It's a joint action between ANSM and the French National Health Insurance. Mahmoud Zoureik co-signed more than 130 articles in PubMed. So I give now let's give you, Mahmoud, you can start the presentation. Thank you.
Thank you very much, Mehdi. Do you hear me?
Yes, we hear you.
Thanks.
Thanks.
I'm pharmaco-epidemiologist and professor of epidemiology and public health at University of Versailles-Saint-Quentin in Yvelines and director of EPIPHAR. I have nothing to disclose related to this presentation. For next slide, please. First, I'll start talking about EPIPHAR with the scientific group of interest, what EPIPHAR is, and then I would like to present you some works on COVID-19 pandemic and some works of vaccine effectiveness and safety of vaccines against COVID-19. Next slide, please. First, what is EPIPHAR? What is EPIPHAR structure? EPIPHAR is the reunion of in 2018 of two pharmaco-epidemiologic units. First one from the French National Agency of Medicine and Health Products, and then others from French National Health Insurance, CNAM, after 10 years history of collaboration. Next slide, please. Somewhat about the mission of, as you know, for ANSM, the French competent authority for medicine, medical devices, and cosmetics, works for safe and effective health products in France and Europe, is involved in pre-market and post-market activity. A unit of pharmaco-epidemiology has been created in 2012, and the missions of CNAM, French National Health Insurance, pays for care of French population regardless of their resource situation or health status. Prevention of upstream of disease is constantly seeking to reconcile the best quality with the best cost. Also, a pharmaco-epidemiologic unit has been created in 2012, and the two teams have been collaborated for more than 10 years. ANSM and CNAM are two public institutions with only public funds. Next slide, please. Next, please. EPIPHAR is the history of collaboration. EPIPHAR, please. Not this one. Next, please. No. I think it's number 6 or 7. Thanks. History of collaboration of many studies before the creation of EPIPHAR, the work program in valproic acid and birth defects and neurodevelopmental disorders. As you know, in utero exposure to valproic acid was associated with an increased risk of birth defects and neurodevelopmental disorders, with a dose-response relationship. Risk of neurodevelopmental disorders, more particularly increased during the second and the third trimester of pregnancy. In 2018, European Medicine Agency announced stroke restriction. Next, and thanks to this restriction and this study, more than 80% drop of '21. Another study concerning cypertron acetate and risk of intracranial meningioma, a work program about oral combined contraception, a study about Assure with permanent sterilization device, and a study for human papillomavirus vaccination and risk of autoimmune disease. Next to this study, it's clear that we need to empower pharmaco-epidemiological study for the decision making for health authorities. Next slide, please. Next, please. Do you hear me?
Yeah, we hear you, Mahmoud.
Thanks. Next, please.
Is
same slide. Please, next slide. Yes. Just before, please. Yes, thanks. EPIPHAR has a scientific political valorization with 33 public reports available in the internet website of EPIPHAR. EPIPHAR has published more than 60 articles in peer-reviewed international journals such as JAMA, JAMA Ophthalmology, PMG, blood, hypertension, etc., etc. Also, EPIPHAR is not an academic structure. EPIPHAR uses research tools for its mission and for its studies. For scientific, we publish a scientific paper for scientific credibility and visibility of EPIPHAR. EPIPHAR also organized the Congress of Pharmaco-epidemiology on public decision. Please save the date of 3rd of June 2022 for the next e-Congress of EPIPHAR. Next, please. I think just before. Next. Okay. EPIPHAR also has a strong partnership policy to develop pharmaco-epidemiology in France by funding of research projects and by funding and hospitality of PhD students and postdoctoral students, funding of 2 centers of pharmaco-epidemiology, Drug Safer, directed by Professor Antoine Parent, and Impulse Team, directed by Dr. Marie Webré in Lyon. Next, please. So, EPIPHAR provides independent public expertise in the field of epidemiology of health products to enlighten public authority in their decision making. I think we missed a slide, number 10, which is the EPIPHAR composed for epidemiologists, clinicians, pharmaco-epidemiologists, statistician, data scientist, and data manager. Could you return, please, to the slide before? No? Okay. No, okay. Slide, I think slide 6. No problem. I will move on. No problem. Next slide, please. Next, please. Same, EPIPHAR provides independent public expertise. Then the next one, please. Excuse me, do you hear me, please?
Yes, we hear you, Mahmoud.
It's same slide, Mehdi.
Okay, that maybe there is some delay between what is presented here and remotely.
Okay.
Probably.
Just before, please.
So maybe just wait a few seconds before you
okay.
You talk.
And perhaps precise the number of the slides to be really sure.
So which slide you're willing to present? Which slide you are, Mahmoud?
One second, please. 7.
7, okay. Sorry about these technical problems, but
no problem. No problem. Thank you very much. As I told, the team composed of independent epidemiologists, independent without any conflict of interest, clinician, pharmacologist, biostatistician, and data scientist, data manager. Work program structured in 3 parts. First part about using and the safety of health products in real life with 3 approaches. First approach, drugs with massive use, new drugs with or new indication, implantable or invasive medical device. Population-based approach, pregnant women, children, elderly people, and tools for pharmaco-epidemiology databases on adverse events, dynamic mother-children cohort, and statistical methodology. The second part is about response to alerts and crises. A third part to measure the impact of decision, recommendation, and communication. That needs exploitation of the French national health data systems. I will talk about it next. Thank you. Thank you. To the slide.
Next. Next, please. Yeah, no.
Next, please. French National Health Data System. Next, please. What is it?
Okay. Mahmoud, you have the slide online?
No, it's just the title, French National Health Data System.
Okay.
Next, please.
Yeah, we are on the right slide now. So maybe just wait just few nanoseconds.
Thanks.
Thanks.
French National Health Data System includes individual information on all health care expense in France, including demographic data, birth, gender, residence, date of death, presence of chronic diseases, and date of the first registration of chronic occupational, accident and outpatient medical expense, about medication, description of packs in number of tablets, number of packs dispensed, date of prescription, etc., etc. Data and nature of physician interaction with the procedure, if any, for example, endoscopy, that nature of paramedical interventions, that and nature of all laboratory tests, but without the results, public and private hospital data, admission date and duration of stay, main related and associated diagnosis, procedure, and for costly drugs. Next, please.
You can see the...
Not yet.
Okay, so we're going to measure the jet lag.
Not yet.
That's strange. Maybe it's going to hold the Earth. Can you see the slide?
Not yet.
Not yet. Okay, that's strange.
Okay.
And now?
No, I see you.
It's on the data acquisition is permanent.
Slides now. Okay, data acquisition is permanent from birth to death, even before birth, through data of mother pregnancy, whatever the socioeconomic of employment status. Data are available for more than 99% of the French population, representing more than 66 million of persons. SNDF has been extensively used from our team and other teams for pharmaco-epidemiologic study. EPIPHAR has a permanent direct access to SNDF via regulatory access from ANSM and French National Health Insurance. SNDS is an invaluable tool in pharmaco-epidemiology to measure effectiveness, safety of health products, drugs, or medical devices. We can conduct drug utilization, post-authorization efficacy study, post-authorization safety study, benefit risk assessments, risk management, and signal detection. Next, please. I will move on to the second part of my presentation concerning mobilization during the COVID-19 pandemic and the work program of epidemiologic surveillance of COVID-19 vaccines. Half of the team are working for this program since 2 years. Next, please. A work program dedicated to COVID-19 pandemics is composed for use of health products during the pandemic, risk factors associated to severe COVID-19 on death, including after vaccination, epidemiological surveillance of COVID-19 vaccine effectiveness and safety. We used the SNDS data combined with COVID-19 vaccination database and COVID-19 testing database CDEP. Next, please. I will start by use of health products during the pandemic. Next, please. We conducted a national-based study by extracting data of reimbursement data from SNDF, prescription drugs dispensed in out of hospital pharmacy 4 billion line of prescription for 62 million people affiliated to general scheme with follow-up of more than 140 weeks. We have studied 59 therapeutic classes study, drug for chronic diseases, cardiovascular diseases, diabetes, mental disorder and addiction, antibiotic, contraception, vaccine, preparation products for medical procedure, and other medicine, azithromycin and hydroxychloroquine. Next, please. I will present you some example of use of medicine during the pandemic. First one about the use of health products, antibiotic use. As you can see, the red line is the observed use of antibiotics in France during the pandemic in 2020. In the dotted line is the expected use of antibiotic. As you can see, sharp drop in deliveries of antibiotic during after the first lockdown in France. This decrease continued after the lockdown periods, and it continues also in 2021. Next, please. Another example is about the contraception use during the pandemic. We conducted a national-based study by extracting all reimbursement of oral contraceptive, emergency contraception, intrauterine devices, and implants. The study reflects the impact of COVID-19 pandemic in the use of the main contraceptive in France over a whole year and a half. Younger French women were impacted most by decrease observed in the dispensing of contraceptive and prescription of long-acting contraceptive use was also the most substantially impacted. The conclusion of this works is ensuring access to contraceptive during health emergency must be a public health policy priority. Next, please. Another example about the use of intravitreal antivascular endothelial growth factor in use during the coronavirus pandemic. That this one is the decrease in use if the decrease in intravitreal antivascular endothelial growth factor dispensing during the lockdown. This may be explained by difficulty accessing ophthalmology department of ophthalmologists and the decision of ophthalmologists to postpone injection during the pandemic period because of difficulty in disease monitoring, which usually include also periodic visual acuity imaging of us. Next slide, please. I will go for the epidemiologic surveillance of vaccine in presenting works about effectiveness and safety. And to simplify the presentation, I will use the names of vaccines by manufacturer names and not by their own names. Next, please. First talk is about the effectiveness of COVID-19 vaccines. We have conducted several observational studies on the effectiveness of COVID-19 vaccine on the risk of severe forms of COVID-19. I will present you two of them. The first one is about the effectiveness of subject over 5019 at a national level. For this first study, we set up a cohort of 11 million of vaccinated persons with Pfizer, BioNTech, Moderna, or AstraZeneca vaccines, matched with 11 million unvaccinated persons, matching variable being age, sex, region of residence. Unvaccinated persons were also eligible for inclusion in the vaccine group when they received a vaccine during the follow-up, even if they had previously selected other controls. We have included about 7 million persons vaccinated with Pfizer BioNTech, approximately 900,000 persons vaccinated with Moderna, and over 3 million with AstraZeneca vaccine with their respective controls. Comparison of the risk of occurrence of severe COVID-19 outcome, hospitalization, death between vaccinated and unvaccinated persons were performed using multivariable Cox models for each vaccine. Analysis by different follow-up windows, duration of follow-up, and calendar period were performed. Next slide, please. Here I present you the full effectiveness of COVID-19 vaccine. Vaccinated person had at least 91% lower risk of hospitalization for COVID-19 compared with non-vaccinated persons. 91% for Pfizer BioNTech, 95% for Moderna. Similar results were observed and found for in-hospital deaths. Next slide, please. We also we have analyzed the full effectiveness according to the duration of follow-up. The results showed that the reduction of risk of hospitalization persists at least up to 5 months follow-up, which was the maximal duration of follow-up available in this study. Next, please. Analysis of full effectiveness undertaken at the time of circulation of the first wave of Delta in July, August in France. It showed also a reduction of the risk of hospitalization related to COVID-19 in vaccinated group compared to unvaccinated, non-unvaccinated group. You can mention that our study didn't address the COVID-19 vaccine effectiveness against non-severe COVID-19, such as asymptomatic or mild COVID-19, which didn't lead to hospitalization. The study don't address also neither the effect of vaccine booster nor the effectiveness of vaccine against the Omicron variant. A revised article reporting all the results of this study is submitted in a peer-reviewed journal. Next, please. Results of the vaccine effectiveness of Pfizer and Moderna vaccine conducted among persons aged 75 years and older, data available earlier in 2021 were published in the journal Vaccine. Next, please. The second study in the effectiveness vaccine effectiveness was conducted focused in Janssen vaccine. we have included approximately 700,000 persons, which represented 94% of vaccinated with Janssen. We constructed a matched cohort of participants aged 55 years or older, vaccinated with either Janssen vaccine or Pfizer-BioNTech vaccine. Participants of the two groups were individually matched according to age, sex, residence, and date of full vaccination. Each participant was followed up from the day of vaccination or full vaccination to until hospitalization within the endpoint or death or the date of the end of the follow-up on August 31. A person vaccinated with Janssen had 5-6 times higher risk of hospitalization for COVID-19 than those vaccinated with the Pfizer-BioNTech. On the basis of these results and according to Pfizer effectiveness from the same, we calculate the absolute effectiveness of 59%. Our study showed that a second dose of was needed in person who received 1 dose of Janssen. This result was published last week in the German network. I will move on to safety of vaccine. Next, please. About safety of vaccines against COVID-19. Next, please. Next, please.
Maybe just a lag. There's some latency, maybe, Mahmoud, again.
Okay, thank you. Several work about cardiovascular outcomes. First one, myocardial infarction, stroke, and pulmonary embolism after Pfizer-BioNTech in subjects aged 75 years and older. Signal, as you know, signal of hypertension and cardiovascular thromboembolic or hemorrhagic events have been reported after receiving COVID-19 by Pharmacovigilance Center. Before we conducted a Pharmaco-epidemiology study to assess the short-term risk of hospitalization for these outcomes. Acute. Next, please. This population used the SNDS link to National COVID Vaccine Database. We conducted, before please, we conduct, undertook. Yes.
Oh, il y en a pas.
I hear you. This population-based study used French health data with person comparison using a self-controlled case series method specifically adapted to cardiovascular event-dependent exposure and high event-related mortality. In fact, we have published a paper detailing this particular extension of the self-controlled case series method designed in the Journal of Statistics and Medicine earlier this year. Over the observation period, about 1,000 persons aged 75 years or older were hospitalized for an acute myocardial infarction. 17,000 of ischemic stroke, 500 hemorrhagic stroke, and 7,000 pulmonary embolisms. Approximately around 50% of patients have received at least 1 dose of vaccine at some point during the same observation period. Next, please. We didn't observe significant increase of any cardiovascular event of interest in the 14 days following the first or second dose. Next, please. This nationwide study was the first to evaluate the risk of severe cardiovascular events associated with Pfizer-BioNTech COVID vaccine at entire population level. Findings don't support an association between Pfizer-BioNTech COVID with an occurrence of serious cardiovascular events. Next, please. A research letter in the JAMA has been published about this study. Next, please. And this is the methodological development about modified self-controlled case series method adapted to this context. Next, please. Similar work has been conducted in adults aged 18 to 71 with all available vaccines in France, not only for Pfizer-BioNTech. As you know, about July 20, 31 million of persons have received in France had at least one dose of COVID vaccine. We studied the same outcome: acute myocardial infarction, stroke, and pulmonary embolism. Next, please. We used also national SNDS combined with coupled with the national COVID vaccination database, and the same methods was used, adapted self-controlled case series methods. Next, please. For the results for this population, the number of cases is a study of Pfizer was the most widely, which is a most widely used vaccine, and for the longest period is the highest. The number of cases was the lowest for Janssen vaccine, used less and later in France, especially for hemorrhagic outcome, hemorrhagic stroke. Next, please. Incidence of various serious cardiovascular events was not increased in the 3 weeks after the first or the second doses of mRNA vaccines, Pfizer, BioNTech, or Moderna. Next, please. In contrast, the single dose of Janssen vaccine was associated with the slight increase in incidence of myocardial infarction during the first and second week after vaccination, with a relative incidence of 1.57 and 1.75. Also, incidence of acute myocardial infarction and pulmonary embolism increased in the second week after the first dose of AstraZeneca. The relative incidence of pulmonary embolism was 1.41, and for myocardial infarction, 1.29. Next, please. Findings of this study are reassuring about the short-term cardiovascular safety profile of mRNA-based vaccines in adults under 75 years of age. In contrast, adenoviral-based vaccines appear to be associated with slight increase in the risk of myocardial infarction and pulmonary embolism. Next, please. Maybe I have time to present myocarditis and pericarditis. I try to make it shorter.
Yeah, please. Yeah, Mahmoud, we are really late, so maybe just go quickly on this specific topic, which is very important. So,
okay.
Just few minutes, and we conclude.
Next, please. Next slide, please. Next, please. We conducted a study of association of COVID-19 mRNA vaccine with myocarditis and pericarditis. We conducted a matched case control study of each condition from the entire French population aged between 12 and 50 years from May to August 2021. Next. We used the SNDS data which collect individual, next please, records of hospital discharge and COVID vaccination and testing databases that can be linked. Next, it's toujours the same. For myocarditis, I think it's there is a lag between this slide and the next ones. Okay, for myocarditis, we included 919 cases which were matched with 10 controls. Those cases were more frequently male and had a median age of 26. You can see the odds ratio of being exposed to the vaccine within 7 days in cases under 30 years of age. Starting with male, the first dose of each vaccine has an odds ratio of around 2, and the second dose have an odds ratio of 11 of Pfizer-BioNTech and 80 of Moderna vaccines. Odds ratio are also important for girls and women. Next, please. For pericarditis, the conclusion that the odds ratio is less important than for myocarditis, but still high for the second dose for each vaccine. Next, please. This one, some estimate of excess events relative to administration of each vaccine. For male, for example, for Moderna vaccine in blue, that's myocarditis attributable to Moderna vaccine is 13 for 100,000 doses. It's around 4. It's around 4 for Pfizer-BioNTech. Next, please. Next.
Which we are what? Mahmoud, do you hear us?
Yeah.
Okay. On which slide you are?
It's the figure, not the slide of conclusion. I will summarize. We provide population estimate of the risk of myocarditis and pericarditis associated to vaccines. We found an association with both mRNA vaccine with 7 days of receipt, but not for longer delay of exposure. We find that the risk is stronger in adolescent and young people under age of 13. Following this study, French authorities have advised against use of Moderna vaccine for those under the age of the 30. The last slide, please.
Yes, we're on the conclusion.
In conclusion, I don't see the slide. In conclusion, we carried out several pharmaco-epidemiology studies during COVID-19 about use and risk of health products, about risk factors of severe COVID-19, about surveillance of COVID-19 vaccine. The results of effectiveness showed that strong effectiveness, more than 19% vaccine to reduce severe form of COVID-19, and we don't observe association between Pfizer-BioNTech vaccine and serious cardiovascular events, Moderna vaccines and serious cardiovascular events. In contrast, adenovirus vaccine were associated with the small and slight excess risk of myocardial infarction and pulmonary embolism in 50-75-year-old people. Association between mRNA, RNA and vaccine and myocarditis and pericarditis in 19-50-years-old people, and higher risk in young women and with Moderna vaccines. Thank you very much. I'm sorry to, I'm happy to respond to your questions.
Mahmoud. So maybe we go quickly to the questions from the audience. Okay, coming. Yes, welcome back. So yeah. Okay, we have a question. Mahmoud, it's how the pharmaco-epidemiology could evolve in the future based on this experience.
The use of pharmaco-epidemiology is increasing since several years. And I think that the real-world study has become important in providing evidence in treatment effectiveness in a clinical trial. Pharmaco-epidemiology study have many strengths and also many weaknesses. If we take the example of vaccine against COVID-19, with the exception with the early results of randomized clinical trials, almost all scientific knowledge has been generated from pharmacovigilance and pharmaco-epidemiology study. I think that pharmaco-epidemiology study could provide information complementary to randomized clinical trial, complementary to pharmacovigilance data. They can provide information in long-term safety, particularly pertaining to rare events, effectiveness of drugs, and the large heterogeneous population. I hope that I respond to the question.
So much, Mahmoud. So maybe we go to the next presentation. So I let Jean-Michel to introduce the next speaker.
Yes, the next presentation.
Thank you, Mahmoud.
Thank you. Thank you, Mahmoud. Thank you very much. Yes, and we introduce the next presentation, communication and transparency, a twin side of public health policy strategies. And I'm very glad to introduce Rosemary Ratignier, the director of communication department in regulatory French agency. And Rosemary has a background from communication and consulting agencies, And at the ANSM, French agency, they conduct the communication and information policy in permanent relationship with stakeholders and in line with French agency commitment to dialogue, transparency, and what is important, risk management. Rosemary, you are on the air.
Thank you, Jean-Michel. Thank you very much. Good morning, everyone. For the duration of the COVID-19 crisis, we have been in constant communication with our stakeholders and with the general public to keep them updated with all the latest developments next. A proactive approach was adopted, allowing us to open dialogue with the public by asking for feedback and sharing information. In line with our policy of transparency, a number of public online meetings were organized, such as today's. Healthcare professionals, patient associations, mainstream media journalists, and others were encouraged to participate. We provided them with easy-to-understand answers, which we published on our website, social media accounts, and YouTube channel. Next, thank you. Just over a year ago, when the vaccine was rolled out, there were a lot of concerns from the general public and from healthcare professionals. In order to allay their concerns, we issued regular updated guides containing information on the potential side effects of each vaccine, along with instructions on how to report them on the Health Ministry website. We also encouraged vaccination centers, pharmacies, and doctors to print and distribute the information guides on side effects to maximize public awareness and reduce fears and vaccine hesitancy. Next, thank you. So as Mehdi told to you previously, we worked on guides. Then you can see guidance for healthcare professionals. Next, please. And then guidance for the general public. Next. For the past year, ANSM has been publishing weekly updates on the latest vaccine information, including reporting side effects. All related information on COVID-19 is available to download and share on the ANSM website, as well as on our social networks, Twitter and LinkedIn. Next. I would like to thank you for your attention and remind you that you can subscribe to our newsletter on our website for regular updates. Thank you.
Do we have some question? I remind that you can ask your question by the chat, either in English or either in French language. Is there any question?
No, next presentation.
No, I think that one question. Yes, question. What kind of difficulties for communication have you met in this crisis context, Rosemary?
Communication in such a context is a hard job because we really need to share very sensitive data. We have a lot of data to analyze, and the aim is how can we communicate and how can the people can be sure about the vaccines, and it's a very, very hard job to do it.
Thank you. So I think we have to go on because we are a little bit late. And I'm also glad to introduce Annette Christine Stark for the next topic. The next topic is real-world data in the European decision. And Annette is a medical doctor specialized in neurology and also has a PhD in neurostatistics. and she has been working as a Danish member of PRAC, Pharmacovigilance Risk Assessment Committee, for the past 3 years. And I remind that Denmark is a PRAC rapporteur for COVID mRNA vaccine Moderna. Annette, I give you the floor.
Thank you very much. Yes, so this lines up very well with the two previous speakers. I'm very happy to see that. So thank you for the opportunity to present at the ANSM. I'm honored, and I hope that the EU Pharmacovigilance Risk Assessment Committee, the PRAC, will be able to visit your beautiful country this summer in relation to the 2022 French Presidency of the Council of the EU. This is not the first slide that I'm seeing, but maybe, yeah, that's probably. I'm going to talk to you about the use of real-world data in the European decision. I'll do that as I have acted as the PRAC rapporteur for the two products used in the COVID setting, namely the mRNA vaccine and Moderna Spikevax and the ibuprofen. So, the slide is a pre-COVID slide describing some basic principles of the PRAC. It's not a new slide, but it's nonetheless highly relevant as how to work with the translation of data into recommendation for use of drugs. And the basic principles are a risk-adjusted approach, that's rational use of resources, clear roles and responsibilities, and reduced administrative burden. And further, transparency is the key to involve stakeholders, provide insight, and hence strengthen the trust. I'll now go through the handling of two signals in the PRAC with focus on not only use of real-world evidence, but how RWE in these two specific settings was used uniquely to solve some of the new situations that COVID continuously have put us in. And when we think about it, reality always has and will do in the future too. I'll first focus on the mRNA vaccine and the risk of myocarditis, which you have heard of previously in the session, and then broaden the perspective to ibuprofen and the risk of exacerbation of infection from 2020 to further bridge to the rest of the use of the real-world evidence. So on the next slide, please. The signal of myocarditis was started in June 2021. And myocarditis is a non-infectious inflammatory condition of the heart muscle. It's the leading cause of disease-related death among seemingly healthy young men in the European setting, and that's even pre-COVID. Also, as you probably know, myocarditis can be caused by COVID itself. On the next slide. This is to give you a quick overview of the signal handling. In July 2021, the text of the product information for the mRNA vaccines were updated and direct information was sent to all the doctors of the EU and the DHPC, that's a Dear Doctor Letter of Information, was also sent. However, in the fall of 2022, we were contacted by researchers flagging real-world evidence indicating further specification of the risk of myocarditis with the mRNA vaccines, leading to a further update of the authority-approved description of a drug plus the patient information based on the French and the Nordic epidemiological studies. On the next slide. So this is unpublished data presented by the researchers to the PRAC. And here I would like to highlight the fact that it was unpublished data. It went really fast and there was an unprecedented collaboration with the researchers who were willing to share at a very early stage, which we are very grateful for. So it's based on the EpiFair case control study, which you have seen previously, and also the Nordic cohort study. And they have different follow-up periods, etc. I will not go into the details of this. On the next slide, please. Here's some extracted data from the EPIFAIR case control study. And what you should note is that it's males from 20 to 29. And as Mahmoud said, we have the odds ratio, but also the excess number of events is even more, how to put it, user-friendly, especially comparing between different studies. Here it's per million doses, but in the product information we used excess numbers of events per 10,000, a number we would not have had without real-world evidence studies. And you can easily see, as Mahmoud said, that there's an increased risk for the younger males. On the next slide, please. The same for the Nordic cohort studies based on a population of more than 23 million. The age groups were a bit different. Incidence rates are still higher in the younger group, not unexpected, as that is the natural cause of myocarditis. But excess numbers of events tells the same story, but as mentioned, they're easier to translate to the user. That could be the healthcare professionals, but also the population itself. On the next slide, please. So in summary, across different populations and study designs with different strengths and limitations, a robust increased risk of myocarditis following spikevax has been observed in males younger than 30 years old. And an unprecedented cooperation between researchers and the PRAC enable a timely communication of the risk via product labels. And again, willingness of the individual research groups to share data ahead of publication. We're grateful for that. On the next slide, please. So this is the product information adopted in October 2021. This slide is to give you an introduction to where we are. This is the 4.4, the warnings and precautions, which were identical for the two mRNA vaccines. On the next slide, you see, yeah, exactly. You see the information to the patient, which is also identical. And then on the next slide, you see the 4.8, where you can see the number, the extra cases of myocarditis in the 12 to 29-year-old males per 10,000, and also for the 16 to 24-year-olds. So these numbers are directly taken from first the Nordic study and then the French EPIFAIR studies. Thanks. Yeah, and this is the same for the Comirnaty. And on the next slide, you can see that the two that here the texts are comparable and only the numbers are different, and you can see that it was a way to communicate that it seemed like the risk was greater for the spikevax than for the Comirnaty. So, so if you think about the first slide, this was a risk-adjusted approach, rational use of resources, clear roles and responsibilities, and a reduced administrative burden. Next slide, please. So I'm briefly going to touch on ibuprofen because I think that's a beautiful story too. On the next slide, you see the tweet from March 14, 2020, from the French Minister of Health regarding a concern of possible aggravation of COVID-19 infection with the use of ibuprofen. Next slide, please. At the time of the signal procedure was already ongoing. However, the tweet and the following concern regarding ibuprofen accelerated the process, as I visualized in the next slide, please. This is an outline of a normal signal procedure with detection of the signal, then validation, confirmation, analysis, assessment, and recommendation. Next slide. Yeah, this is the outline of a norm. Yeah, this is obviously far too busy slide, but it's just to give you a visual impression of the complex but planned situation we were in regarding the signal of exacerbation of infection with ibuprofen and ketoprofen at the start of the year 2020. I'll not go into any further details, however interesting. The point of the slide is to illustrate the signal procedure was well underway at the time of the tweet and the additional concern of possible exacerbation of COVID-19. Next slide, please. So the triggered the signal in 2019 due to several studies indicating a risk. And this data was processed during the signal assessment. Majority of this data was on bacterial infections, so media and tweets brought attention to the signal and prompted acceleration due to the new concern. And DKMA initiated collaboration with Anton Pottegaard, from the University of Southern Denmark, SDEU, to perform a retrospective study to investigate the risk of viral infection, more specifically influenza. And preliminary data was included in the assessment and later published. As you can see here. Next slide, please. So this is the EMA press release already 4 days after the tweet. And EMA, yeah, I know you can't read it, but here already EMA emphasizes the need for the collaboration. And EMA literally states, EMA highlights the need for epidemiological studies to be conducted in a timely manner. So what I wanted to illustrate with this story is the last part of the initial slide, that transparency is the key to involve stakeholders, provide insight, and strengthen the trust. Next slide, please. So a rational use of resources is increasingly evident, and clear roles and responsibilities. That's both local authorities versus the EU collaboration, and reduced and administrative burden. I believe this can be done with communication, communication, and communication. And also the transparency part to involve stakeholders, but also be aware of the public and the new players in the game. That's also SoMe. And provide insight early and separate communication, especially on ibuprofen, and strengthen the trust. And this is especially important with the vaccine credibility. So in conclusion, real-world evidence is here used to bridge to the product informations. The experience of collaboration between the researchers and the regulatory authorities during COVID-19 shows that timely conduction of epidemiological studies can contribute valuable information regarding complex information derived from epidemiological real-world studies to be condensed into recommendations that may impact the treatment of patients or even be directly implied in the decision of whether or not to vaccinate. Again, thank you to the researchers for their willingness to collaborate.
Thank you very much, Annette. I think we have 2 or 3 questions. The first question about communication and illustration of the level of risk. because people might have some difficulties to visualizing a risk when expressed as 1 in 10,000 or more, or very similar. And can comparative risk, as risky as swimming in a pool, help with the understanding of risk magnitude? What do you think about that? Of course, if you want to cross a motorway with a bag on your head, that speaks to everybody. But how to express the very low risk?
I think it's difficult to give a picture like that. It's difficult to give a picture like this is like swimming in a pool, or but I think what we ended up doing, using the excess risk, a number for the excess risk, I think that somehow, at least for me, illustrates it better than a confidence, like a classical way with an incidence rate ratio. I still think it's still translating into this is 18.8 more cases per 100,000 than expected. I think that's explainable. Thank you.
Okay, a second one, Annette, how the national recommendations have an impact of the European assessment, and so the interaction between the national level, European assessment. What is your point of view about that?
I think the national input is the most important part of the European community. I think that's the essence of it all. That every European country gets its say, and we get all the different inputs, and hence get a much better output that way. Thank you.
Okay, and the last one, I think it's a bit more tricky question, because in the context of pandemic, our European agency has evolved, signal management, communication. What can you say?
How has the I'm not sure I understand the question. How has the national
how do you judge the evolution after the management of pandemic, both at national and at European level? What do you think about evolution?
Okay, that's an easy question. Because before the pandemic, we were in the Stone Age, and now we have evolved immensely, and the collaboration has evolved immensely, and I think the pandemic has taught us a lot. Some of it better than others, but I'm sure we will come out of it strengthened. Thank you.
Okay, thank you for that nuanced shade of hope for this end. Thank you. Mehdi, for the conclusion of the two sessions today?
Yeah, so we go for the conclusion. So we wrap up the what has been talked so far. So what we could see here is that this first session demonstrates that real-world data contributes significantly to prove the efficacy and safety of COVID-19 vaccines. We could also see that there is some similarity between the organization put in place in France, in Norway. We could see also, we have been informed how really each report is analyzed on both sides, on Norway side, France side. we could also see that there is a strong relationship between pharmacovigilance and pharmaco-epidemiology, because we are starting from a sample of, from a signal detected from the pharmacovigilance and then a confirmation by the pharmaco-epidemiology. We have seen so far some good examples of this synergy. Myocarditis, we have seen also the example of menstrual disorders. So what we could say that it's there is 3 key points of COVID-19 surveillance. The first one is the rapidity, reactivity. This is the first key point. The second one is the agility. And the third one is the transparency. So that what summarize, I mean, the first part of the first session. Jean-Michel?
Yes, and for the second session, I think it's complementary with that told us Mehdi. It's the importance of the collaboration of health insurance systems, regulatory agencies, and also academic institutions. It's a very fruitful collaboration. And the importance to empower pharmaco-epidemiology study to help health authorities for making decision, public health decision, uppermost in emergency situation. And I think that, there is not an old area and a new area. We still have some collaboration and complementary between more classical way of knowledge with trial, clinical trial, and now with pharmaco-epidemiology. But sure that in a pandemic, such a pandemic situation, with a very emergency situation and a very evolutive situation, pharmaco-epidemiology is a really new and very important tool. It's my conclusion. And with communication and transparency to assure the go and back with population and with patients.
Thank you so much. So the first session is end for now, and we welcome you for the next session at 2 p.m. Thank you so much. Have a nice lunch.
Good afternoon, everyone. I am Vincent Gazin, Deputy Director, Europe and Innovation at ANSM, and I will moderate this session together with Jean-Michel Arras, who is French CHMP and CAT member. We will be helped for taking your questions from the chat by our ANSM colleagues, François Queneau, Marie-Joëlle Jabaji, Noronsoa Rassolon-Dramanitra, and Marie-Caroline Pesquidou. Thanks in advance to them.
Thanks to them. And as emphasized in the pharmaceutical strategy for Europe of the European Commission, secure and efficient access to health data is a crucial key point to fully exploiting the huge potential of new technologies and digitalization. Industry and regulators require access to data, and through a robust European Union-wide data infrastructure to support innovation and safety patient. And an interlinked system that gives access to comparable and interoperable health data from across the European Union would be a real multiplier in terms of research, regulation, and evidence generation.
Initiatives across European member states are the bricks for any European construction and networking. We are both, Jean-Michel and I, delighted to welcome you this afternoon to a session dedicated to works and local initiatives that allowed in several member states creation of networks enabling the collection and the analysis of real-world data. Experiences are coming from Denmark, Italy, or Spain, and will illustrate the complexity, but the success as well, to set up collaborations between institutions, to generate good quality data, to appropriately use digital tools. As presented this morning, COVID-19 pandemic was a major impulsor of data generation, and some of presentation are addressing it, but not only, and other fields will be presented. At the end of the session, we will welcome the integrative initiatives led by the EMA in order to consolidate and harmonize the use of real-world data in the regulatory decision-making process. Let me introduce the first speaker, Anton Podkart, Professor of Pharmaco-epidemiology at the University of Southern Denmark and Head of Research at Hospital Pharmacy Funen, Odense University Hospital. During the COVID pandemic, Anton and his colleagues have worked extensively with Danish regulators to support their decision-making around the use of medicines during the pandemic. He is going to tell us about academia and regulator collaboration. Please, Professor Podkart, you have the floor.
Thank you. I hope this is working. It seems it's working, although with a major echo, and that's now gone, I think. Otherwise, I have to kill the sound on my end. Thank you, and thank you for inviting me and for a chance to present some of the findings, or rather the approach that we've had to setting up real-world and real-time, or almost real-time, data generation to support regulatory decision-making during the COVID-19 pandemic. I hope you will be able to, there's my slides, wonderful. I'm representing the University of Southern Denmark, and next slide, please. Have to say next slide, please, a few times because I cannot control the slides myself. One of the key reasons I think we've really seen an interest in what we can do with the Danish and Scandinavian data sources is because we were able to, as a case example, present the findings on the AstraZeneca Oxford COVID vaccine and VIT. This was something generated, of course, by spontaneous adverse event reports and discussed widely, but we could very quickly in the Scandinavian data landscape provide a quantification of how this phenomenon affects people receiving the vaccine. So this would be the usual example. I think this has sort of really highlighted the value of this. But I would like to, in this presentation, seeing as this is already over and done with that particular vaccine and that study, try to highlight some overarching thoughts on why we came into a position where we could do a study like that. And more importantly, I will end with what this could mean in terms of how we leverage academia-regulator collaboration in terms of supporting regulatory decision-making in the future. Next slide, please. So the question is, where did this all start? Next slide. So this, to me personally, was the initial stepping stone. This is a news bulletin from 2015, and this is in Danish, but this says your heart medication can kill you. This is based on spontaneous adverse event reports that the Danish regulators had received, seeing as someone who switched from warfarin into a generic warfarin product, so generic substitution, they had submitted reports of experiencing bleeding events. So this was shared, and it was really shared major news outlets, and in particular a few Facebook posts who created a lot of fuss about the question on whether this was in fact true. Next slide, please. So we tried to address this issue because this is a clearly formulated hypothesis. Is it dangerous in this particular time zone to switch from this drug to the generic formulation of the same drug? So we published this study. Next slide, please. In comparison to what you can do with standard pharmacovigilance data, that can raise a hypothesis, and it did, but we can actually address it in full, not only providing the number of events, as you see in the second column here, mapped according to whether the single individual had recently changed to the generic warfarin or had continued using either the original or the generic version. But we can also map how many people did this without experiencing an event. And from that, we can estimate rates and relative rates, or here hazard ratios. And as you can see from the rightmost column, there is absolutely no evidence that there is an increased risk of bleeding. So from the leftmost column, the number of events, it's worthwhile noting that of the 53 different events happening after a switch to a generic, we saw, I think, 19 of them being reported to the regulators via spontaneous adverse event reports. But of the about 5,000 events during continuous use of the branded drug, 0 reports were submitted, because this is a standard thing. We know people bleed. It's only in the case of a switch this triggers suspicion and reporting. Using real-world data is free of similar limitations, and we can address such a question. So this sounds like a success. We could provide a strong answer to whether this is a problem or not. Next slide, please. However, as you can see from this, the news bulletin is from May, and we submitted the paper in August, and we had it accepted ultimately November 23rd. Clearly, this is completely useless. From a regulator point of view, they would have sometimes during the afternoon of the very same day the bulletin came out, they would have to reach a decision, should we suspend or should we not suspend the generic substitution of warfarin products? And of course they did. And while we end up concluding there is no major risk going on, this is not in disagreement with the decision they reached in real time to stop the potential problem while waiting for a characterization. Next slide, please. In 2018, we had a new potential crisis, and by random chance, I had just had lunch with someone working with the Danish Medicines Agency, and this came out 2 days later. We just talked about the fact that if something happened in the future, we should probably collaborate much closer and try to arrive at something much sooner. Next slide, please. So when this news broke, we very quickly established a data set with the support of regulators. So we could speed up the approval processes. We mapped out the fact that this potential NDMA contamination of antihypertensive valsartan apparently affected about a quarter of the products on the Danish market, and another quarter potentially, remaining 50% probably not affected. Next slide, please. This allowed us to do an assessment of this phenomenon, and as you can see from the circle here, this was published in the BMJ under the fast-track submission system. In practice, this means that from the day the news broke, that is worldwide problem with NDMA contamination published on media, from that day, it took about 5 weeks before we had a preliminary response to regulators, and an additional 2 weeks, so 7 weeks in total, before we submitted the paper, and 10 weeks in total before the paper was published online. This means that instead of having a 6 or 7 months later commenting on whether a decision was correct, we could now, in parallel to the decision being made, support the regulators and say they could lean on the fact that data would probably come very soon, and that could be part of the decision-making process. Next slide, please. Corona came about. So this 2015-2018 experience and the very close collaboration with Danish regulators, including the regulators having learned that they can actually utilize such data, made it much easier to collaborate now that we really had to. Next slide, please. So we established the DAC-COVID. DAC-COVID is an sort of experiment trying to address hypotheses related to medicines, led by the Danish Medicines Agency, with the steering committee, including more or less any Danish regulatory body and also Danish patients and other interest organizations. And I was appointed project lead. I'm not working with the Danish Medicines Agency, but University of Southern Denmark. But on behalf of the Danish Medicines Agency, I led this project. Next slide, please. The overarching aim was to provide, as stated here, rapid analysis of any questions related to the use of medicines in COVID-19 with potential important public health impact. Next slide. So we gathered all this data, mainly, of course, SARS-CoV-2 PCR test results, and linked it to all the usual data sources we have in Denmark: prescription fields, hospital admissions, death migrations, lab data, ethnicity, etc. So early in the pandemic, we had about 20-hour lag on SARS-CoV-2 PCR results and about a week lag on prescription fields. So twice a week, we had new data fitted into the system, and we every week sort of pulled all the other registry data in. So having a almost real-time data set covering the Danish pandemic as it developed. Next slide, please. So once we had this, we described this, of course, we sent out this paper and also distributed our description of the data sources via other media to potential collaborators. And if you would like to take a look at the technical details, it's here. Next slide, please. So we very clearly said to every Danish potential collaborator saying we are open for business. For technical and legal reasons, there was only one version of the data set to begin with, residing with my group, but this is not an attempt for me to monopolize this, or our group or the Danish Medicines Agency to monopolize the data, quite the contrary. So we very clearly said to everyone, we are open for business if you have a hypothesis. And this is updated a little more than halfway through the project, with hypotheses being submitted by research groups all over Denmark. The stars here corresponds to, I guess, all the major epidemiological groups in Denmark. They submitted hypotheses to us, and in a collaboration with an expert group, which I led, including epidemiologists and statisticians and clinical infectious disease specialists and etc., etc. From all over Denmark, we collaborated on setting up these studies. Next slide, please. The workflow was that they submitted a hypothesis to the steering committee. Once the steering committee approved this, mainly evaluating whether this has to do with medicine and is of public health importance, the submitter, the external group, met with the expert group. A protocol was developed. The protocol was registered in the EU PAS because that was a requirement or an ask by the EMA via the Danish Medicines Agency. We then conducted all the programming. We prepared a note for regulators and submitted the paper. Note here that the regulator note is bolded because the main deliverable for us was in fact a regulator note sent to the Danish Medicines Agency and via them to the European colleagues in EMA. So the data we generated was submitted to the regulators before being submitted to journals. So almost real time, I'd say, input to the regulators. Next slide, please. By now, we have completed, I think, these 16 studies, 5 of them while waiting for the data to accumulate. So we did a few studies on influenza patients, some methodological work, and then 11 hypothesis evaluating studies. All but one are long since published. Next slide, please. As you will likely receive, you cannot read this, but you will likely receive the slides. So here's a list of all the projects, the leaders of the individual projects. We have titles and where it was published and link to that publication. So all of this is made available. And since it's all COVID-related research, I think all of it is open access. So thank you. Next slide. A few highlights of what we have been doing within the portfolio. I want to highlight this one because it's very important for us that what we did was not only fast, quick, and in the hands of the regulators as soon as possible, it also needed to meet highest methodological standards. And while waiting for the data to accumulate and building this platform, I worked with colleagues, including from the EMA, Xavier Kurz, but also other established colleagues within European pharmacoepidemiology to provide guidance. This is endorsed by the International Society of Pharmacoepidemiology as sort of a starting point for methods issues specific to the SARS-CoV-2 pandemic. Next slide, please. Other sort of early papers that came out was sort of characterizing what is the data material at hand. For instance, here the first 11,000 cases. Next slide, please. This highlighted, for instance, what is the sort of composition of people dying or being admitted or not requiring hospitalization overall according to age and according to age and comorbidity. These data seems almost ancient or somewhat trivial by now because we know this is what it looks like, but we most certainly didn't know this very early on in the pandemic. It was, for a descriptive paper, this generated quite a lot of attention because this was really something that was a high demand by regulators, but most certainly also by the lay media and the public in general, having a transparent sort of display of how is the pandemic influencing us while we progress through it. Next slide, please. Another example was this one. This is, I think, my favorite example of how we had to rely on data very quickly to sort of make our way through the early panicky-like stages of the pandemic. This particular hypothesis was raised initially by French pharmacologists, and I have been sort of looking into this, and I've actually talked to the people that talked to the French Ministry of Health about this potential issue. This was flagged as a potential issue. However, we all felt an urgency to do something. So once the French Ministry of Health had this potential information, at some point it was tweeted by the French Minister of Health saying we have a potential issue with NSAIDs being dangerous in COVID-19. This was probably overstated a bit. It was a hypothesis at the time and only a hypothesis. But it was published and it was picked up by news media all over the world saying, oh, if you would like to do something yourself, you have to stay clear of NSAIDs. You, of course, have quite many people using NSAIDs. This is a common analgesic. So people were both scared to take analgesic if they felt ill before having SARS-CoV-2 disconfirmed. And those people using NSAIDs on a regular basis were very concerned that they put themselves at risk of COVID-19. Ultimately, this was picked up by the WHO, who issued a warning saying we should really avoid using NSAIDs in COVID-19. This is, to the best of my understanding, outside the purview of WHO, and that warning was also retracted about 24 hours later. And this is certainly not put forward as a criticism of anyone involved in this portfolio. I think it just reflects our sort of common need to act, to do something in the context of the early pandemic. But we need data. We need to provide actual data, whether there is a problem here, of course, because while this is just a hypothesis, it needs to be confirmed or disproved. So we did a study, actually prepared for this study using influenza data. So looking into whether NSAIDs carry a risk in other viral infections. And as soon as we had sufficient data, we did this study in SARS-CoV-2 patients. Our findings have later been confirmed by a ton of other studies. Next slide, please. So as you see from this slide, unfortunately, the layout in this journal is not ideal, but as you see on the rightmost column, the risk of death, hospitalization, ICU admissions, mechanical ventilation, or renal replacement therapy is all very close to the null. There is no increased risk. This is the relative risk ratio. There's really no risk with using NSAIDs. And I think overall, this is a common theme for many of the studies, if not all of them. This has been hypothesis being put forward that has caused quite a bit of alarm, but we had the opportunity to rely on data to actually disconfirm these suspicions, providing reassurance to both prescribers and patients, and of course also regulators. Next slide, please. I will, of course, not provide all the examples because it's 16 papers and then comes the vaccine portfolio. Another example of the use of this data is the chance of looking into post-acute effects of SARS-CoV-2 infection. We, based on data availability from other centers, in particular US data, we had quite some data arguing that there might be risk of sequelae or long COVID, in particular among those early in the pandemic, in particular among those admitted. But the vast majority of patients with COVID-19 aren't admitted. And due to data availability, or rather data not being available, we had almost no data on this very large group of non-admitted COVID-19 patients. And it's realistic they have a different pattern in terms of their long COVID compared to those that were admitted with their initial disease, yet no data. And this data was actually being asked by a Danish regulator, saying we have to prepare long COVID clinics. Based on the data we have, it seems we have to prepare for a surge of patients with long COVID, but we're unsure what this is actually going on, how many, what are their symptoms, and how is this reflected in healthcare data. Next slide, please. And as you can see from this very busy figure, we looked across a ton of different outcomes. What's the risk of myocarditis, heart failure, venous thromboembolism, pulmonary disease, kidney injury, blah, 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 blah. We could, in the Danish data, on this very large cohort of individuals not admitted, that is the majority of patients, see that, well, actually not a lot is going on. We have a slightly increased risk of diagnosis of, for instance, dyspnea, stuff not really well captured in the registries, but there's something going on for those very sort of unspecific outcomes. But for the hard outcomes that we would really need to know at a very short time frame whether we would see an increased rate of heart failure or VTE, blood clots, etc. And we can very confidently reject that hypothesis. There is no major burden of disease following non-admission COVID. That is not to say that there cannot be persistent symptoms and that they can clearly affect quality of life among those that have had COVID-19, even if they haven't been admitted. But from a hospital management public health point of view, we see no surge of patients admitted with acute disease after infection. Next slide, please. So that brings us to the Oxford AstraZeneca vaccine study. This is technically outside the DAC COVID portfolio, but of course, I must say this is clearly a part of the data that has been generated during the Danish data landscape, or the Danish data sort of project with establishing this. And I would like to sort of go through not so much the results because you know these already, but tying it back to the how can we support regulators real time. I would like to give you the narrative of how we put together this study, and then I will tie it to how can we do something similar in the future, should it be necessary. I hope not, but most likely it will be in some shape or form. Next slide, please. So this is an attempt at sort of trying to draw you a timeline. What happens now that we have an emergent crisis at hand? And from a very practical point of view, how does this affect the work stream that we have to set up to provide real-time answers? Next slide, please. So early on, we have the thrombosis reports highlighted in March 10. The first emerging reports out of Austria, Denmark, and Norway. Very quickly, this led to the vaccine being put on hold. And at that time, we are talking days here, the BMJ published a status saying there's an emerging thing here, but please calm down. Next slide, please. This is from the BMJ paper, and I don't, most likely you cannot read it. I'll just highlight the little section highlighted in red in the bottom right corner. This is from an opinion piece in the BMJ, the news section, and it reads, there is no sign anywhere, including the UK, where very large numbers of doses have now been given, that blood clot related illnesses are happening any more frequently than usual. So clearly in the UK, they had vaccinated a lot more people than we had in Denmark and Norway at the time. But as stated, there is no sign, there's no reporting scheme at this point in time that had caught on to something going on. Next slide, please. So skipping a few days ahead of time, you see a paper published in The Lancet, sort of a fast track paper saying, okay, what is the background rates of venous thromboembolic events? Because at the time we didn't really know what happened. So we needed something to compare with. March 15th, many different countries held the vaccine use. And 17th, we had an emergency meeting in the Board of Health, because now we realize this is not just venous thromboembolic events. Something really peculiar is going on. Next slide, please. This is a screenshot, as you can see, of the Danish director of the Board of Health, Søren Brostrøm, in a meeting with this emergency meeting. They established very quickly a forum of epidemiologists, infectious disease specialists, and thrombosis hemostasis specialists, so that Søren Brostrøm could ask us this question: are we facing a new syndrome? And we could put together our expertise in trying to sort of answer this question and provide input to his decision making. Next slide, please. So around this time, it was becoming very clear this is not just a matter of thromboembolism, also a combined presentation with thrombocytopenia. This was published in very many different news outlets. But the key thing to take from this is that while this happened, a forum was built where people were meeting and discussing this in real time. So we had a chance to put together a timely response. And now we'll take a big jump in time. So next slide, please. This was 17th. As you can see from the now fully updated timeline, the 17th was the meeting with the Board of Health. 19th, we saw the preprint describing VIT. 19th, the same day, we discussed whether we should put together an epidemiological study. This is a Thursday, I must emphasize, Thursday. March 20th, by around noon, we discussed, will you put this in motion, and stuff. People were talking to each other, and by that evening at 8 o'clock Friday evening, the Board of Health confirms they will support all the approvals, so we could get a fast track access to the data we need. Saturday 21st, legal contracts are worked out. Sunday, we receive all the registry data. This was a matter of people being pulled in from weekends within the Danish Health Data Authority to set up the data access. March 24th, we provide the first crude analysis of saying we see this many events, and based on background data, this is more than would be expected. The 25th, we formally include the Norwegian data partner, but we had been talking to them since Sunday evening. Drafting a manuscript for 26th, paper ready, and this is forwarded in a crude draft to the Board of Health and via them to European collaborators. PRAC is informed, EMA is informed April 3rd, and by April 9th, we submit this to the BMJ under a fast track scheme. Note here that the 3rd to the 9th were the part where we relaxed a bit, took a step back, redid everything, absolutely certain we got it right before submitting. The key thing is here, not the 9th, it's absolutely the 31st and 3rd. We could provide a timely answer, including a statement from the researchers involved saying this is how we see the data in a very short time frame. Next slide, please. So, and this brings me to my conclusion, and hopefully this will trigger some discussion and questions. Because this is an interesting paper, and it was a very hectic and I think very important time that we managed to, and when I say we, I emphasize we as Danish epidemiologists, including I think researchers from all major epicenters across Denmark, really took a sort of managed to step up and provide input to the regulators when they needed it. But as an afterthought, or rather as something to take from this other than the estimate that the study gave, I think it's important to try to look into how can we do this even better in the future. So the key line here from signal over process to decision is a crude, very, very crude representation of a regulatory decision-making process. So starting off with the signal, there is something going on, and signal question mark, because it needs to be decided at some point whether this is a signal, or rather whether this is a signal that deserves attention and requires some sort of action. Once that is taken care of, then it moves into some sort of process. Someone are now formally looking into data. They are trying to evaluate whether this needs to, what type of action is required, and ultimately we see some sort of decision. This could, for instance, be disbanding generic substitution of warfarin based on the signal that came from spontaneous adverse event reports. So the key thing here for me as an academia, a potential collaborator to regulators, is to see how can I support this process. The rightmost paper, all the way down to the right, is the warfarin analysis I presented earlier on. This is a post-decision evaluation, whether what the regulators did was actually the right thing to do. Most of the time, those papers will provide some sort of assurance or guidance, but they will talk to this phenomenon with sort of the clarity of hindsight, and very rarely they will actually add to something like you should have done something differently. It's a post hoc evaluation. This can be more useful than what we did with warfarin. It could, for instance, be, and we've also done that recently, looking into regulatory changes in handling of opioids. How has that affected the actual use patterns? They tried changing it. Did they succeed? They did, I can add. Such post-decision evaluations can be very useful, but in this case, it's not really. You can also see a paper like the one we did here with AstraZeneca. This is the second to last paper, just below process. Here we have a paper that addresses an ongoing issue and that helps finalize a process. So it's already been disbanded, stopped using the vaccine. We are evaluating whether this makes sense. But at the end of that evaluation, we come and say, okay, here's sort of hardcore data from registries. This can guide your finalization of the process and decision. Even better, I think, and more valuable is the NDMA paper. I set that on top of process because that's not only to help finalize the process, but the availability of data and the promise that we would be able to deliver data very quickly, to a great extent, I think, shaped the process. It allowed regulators to say, well, we will look at this, but we will look at this over a 4 or 5 week period because we know we will know more very soon. So making those data available and collaborating in making them available helped shape the process and make the process in itself more efficient, not only leading to a distinct decision. But I think we can do more than that. I think we can do better, actually. I think we could push towards the X on the upper left corner. The example I've given here is a paper on DPP-4 inhibitors and splanchnic vein thrombosis. These are thromboembolic events in the stomach, in the veins near the liver. You have never heard about this concern. I don't think you have, at least. This was published in a high-ranking journal in a letter format saying we have looked into spontaneous adverse event reports. It seems like there might be a problem with an increased rate of these particular types of thromboembolic events in users of this particular drug class. In this case, we, by random chance, intercepted that signal very early on. And we wrote to regulators, and I don't exactly know how they handled our inquiry, but we wrote to regulators saying there is a potential signal here. If you are considering entering into a process, if you're considering it, please know that we will be able to deliver data on this very soon. So if you are unsure, you can tip to the not necessarily turning into a process, because we will soon be able to sort of supplement those data. And we did very quickly. And of course, again, we forwarded the data to the regulators before submitting the paper. This, in a much larger study, clearly shows there's absolutely nothing going on. And as far as I know, no process has been started. I should not assume the process within the regulatory agencies, so I cannot tell you this is something that would otherwise have been a major thing. Most likely it wouldn't. But as a theoretical example, at least, trying to intercept a signal before the very costly and time-consuming processes are started, it can at times be valuable to sort of look into registry data to qualify a signal, or rather qualify whether something actually is a signal or can be considered a signal. Finally, of course, the arrow to the bottom left, please note this will, of course, be different from all cases. I'm not arguing that we should have been much quicker at doing the warfarin study. They needed to disband generic substitution at the same day this happened. We would never have been able to meet that deadline. They needed to suspend AstraZeneca vaccination, or at least they opted for doing it. We would never have been able to inform that early on. So it's often a given, but we tend to, in academia, focus on doing the sort of final post-hoc evaluation studies. And I think we need to push in our collaboration with regulators for providing much more timely data. And this requires, I think, quite a lot of development in how we collaborate and how we set up fora where it is possible to entangle or join together the work streams that we have. So I look forward to the discussion. I hope I have provoked someone enough for questions to ensue, and I look forward to answering any questions you might have. Thank you.
Thank you very much, Professor Podgaard, for this very interesting presentation where we see the importance of the publications of data in order to concretize and give real ability of data via the scientific analysis and the scientific point of view. And also was interesting the fact that indeed we are talking about real world data, but real world is also meaning real time. And you talk a lot about the necessity to have quickly the data to allow providing it to regulators, for example, and also to communicate to health professionals and patients. And it's interesting because this is a mean to trust in data for health care professional as well as patient, and do not be only the black box of regulators. We have some questions from the chat, and maybe the first question that I would raise is, as you mentioned, you efficiently deployed a collaborative network in a very short time frame. For example, between the thrombosis reports on the 10th of March and the submission in BMG in the 9th of April, there is only 1 month. Could you come back on the key elements for you that allows such velocity, such a short duration of 1 month?
Thank you. Absolutely. I think one key element, of course, is the data infrastructure. We need to have registries that are more or less continuously updated, and we need to have infrastructure, for instance, like the Danish Health Data Authority, that are able to provide data accesses in a very short time frame. Usually, of course, it takes much longer than this, but they need to exist so that we can actually sort of push the right buttons to make things happen. Another, and I think equally important, but sometimes less recognized aspect, is that you need someone who's able to talk to regulators and know what is going on and have a sort of established dialogue. Something like this cannot be built in this time frame if not for the fact that we have a direct connection and we know the issues they are facing, and we are able to talk to them very quickly on and have an established forum for this. In this particular case, this was actually a forum established through this, but I think it was very sort of brilliant move by the Danish Health Authority to establish such a forum. Only days after the news broke, we will put together, I think, 30 or so experts. Finally, and also somewhat under-recognized, This is a setting where someone really needs to show the way in terms of avoiding standard academia trench battles here. This could be a case of who does what and what institution receives this. But this is based, I think, on a nice history of collaboration between Danish centers, academic centers, that it was really not a major issue trying to make, in this case, 5 different research groups work together to provide an answer. So the easy one, we need the data to exist and the data infrastructure, and the less obvious ones, someone needs to take the lead on establishing a professional forum for this exchange to happen between academia regulators, and we need to put emphasis on establishing a good, sound collaboration with all the interpersonal stuff and all the project management stuff that comes with that. It's not just a matter of doing it, it needs to be sort of a professional leadership around this type of collaboration.
Thank you. So this lead, for example, to a second question, but Jean-Michel, maybe other question.
Just a question, because it was interesting. You showed that this process can answer very quickly to a response, because we used to think that scientific timing was very different than mediatic, written, or TV timing. And I think now with the media, but also with the networks, you have plenty of else beliefs, and how to have a prioritization to treat such news in the public. And also there is also a problem to share this access to big data, not only for public academic university institution, but also for private or for patients. How do you see this future?
I think you're absolutely right that there is a huge need to prioritize what particular research agendas are taken, and there's substantial difficulties in making sure who accesses the data. Even in the Danish context, this was an issue, as for legal reasons we could only establish one data set, and it required for us to collaborate on the same data set quite a few different institutions. In terms of priority, I think that is one of the main things we achieve by having a regulator-academia collaboration, because the regulators can focus on what they do best, embed the evidence we create into a public health setting and evaluate how this should inform the regulatory decision-making going forward, and communicating that. Whereas I or my colleagues can focus on what we do best, that is creating a specific number and a confidence interval that goes into that regulatory decision-making process. And while we collaborate very closely on this, I need to emphasize that I am not collaborating with the Danish Board of Health in terms of whether or not to recommend the vaccine or whether the program should be halted. That's their problem. I am only trying to provide the data they need in order to actually meet that requirement or make that decision. In terms of prioritization, just something you mentioned earlier, in terms of trust, I do think this is essential to ensuring the public trust in regulatory decision-making in general, but also, of course, specifically the vaccine program. We see various issues being raised. As just one example, we see a concern, somewhat hypothetical, that the mRNA vaccines can cause Guillain-Barré syndrome, or it could cause appendicitis, etc. Addressing these concerns, even if it's not based on much data to begin with, the concern that is necessary to ensure the trust. Somehow, if we don't mount large-scale studies to look into the concerns that actual real people have out there in the world, it first off gives some sort of black box feeling, as mentioned, and more importantly, I think it somehow conveys a feeling of disinterest, like we as regulators and as medical community don't really want to concern ourselves with these hypotheses. Just the fact that a large bulk of people are concerned over something should, in particular in the face of a pandemic, qualify something to be prioritized. I think from that point of view, one particularly interesting aspect of the pandemic has been that we were able to talk very openly about what we did. So this study hypothesis from some group was submitted. Everyone in this network knew who did what, so we ensured that there were no duplicate efforts. We also had similar thing going on via the EMA. They established a forum where we could talk about what was going on. We had a long list of studies being conducted here and there. And that helped us a lot in terms of prioritizing, because we could, so to say, divide the issues. Clearly, it would be valuable to have replication. So it's not a matter of only one group can do one thing, but having a forum where we can actually coordinate so that we ensure we all serve in the best interest of regulators, but also public. It was really sort of crucial to ensuring that we were able to deliver on the right things in a coordinated fashion. So I really think we need to prioritize these hypotheses simply based on the fact that they are concerns by those people that volunteer to receive this vaccine. And for that reason, coordination is highly important.
Thank you. And maybe a last question, and maybe if you can answer rather briefly, because we are we must respect the timeline. Do you think it is feasible to screen population or registry data for potential problems when new drugs or vaccine enter the post-marketing phase, including hypothesis-free screening?
I think it's a very important and somewhat underutilized aspect of real-world data. We tend to use real-world data to answer one hypothesis at a time, and that hypothesis then have to come out of trial imbalances or petri dishes, mouse models, or clinical observation. But I do in fact think that there is a potential for using real-world data to generate these hypotheses. Of course, with due consideration as to plausibility and multiple testing and reproducing these signals, etc. But the important hallmark of this is that if a hypothesis is generated directly from real-world data, the chances of this hypothesis surviving the translation into having actual patient impact is much higher. And we have demonstrated this, for instance, by doing a hypothesis-free screening of carcinogenic effects, looking at whether any drugs is seemingly associated to any cancer. This led to the discovery that hydrochlorothiazide, a particular compound, is in fact increasing the risk of skin cancer, something that has been replicated, I think 10 or 15 times by now in other studies. So yes, I think we need to look at new and creative way of using these data sources in order to sort of realize their full potential.
Thank you very much. We are going to pursue on the second presentation, and I let Jean-Michel present the second speaker.
And thank you, Professor Podgard. Thank you. And for the next speaker, I'm glad to introduce Professor Gianluca Trufeo. He's an MD and PhD in clinical pharmacology, and he is currently working as full professor of pharmacology at the University of Verona. He is a member of the Scientific Secretariat of Post-Marketing Surveillance Office of the Italian Drug Agency. He is a scientific coordinator in the Italian Society of Pharmacology, working group of Pharmaco-epidemiology and Pharmacovigilance. He is member of the steering group of advisory board of the International Society of Pharmacovigilance. He is involved in the European network of Center for Pharmaco-epidemiology and Pharmacovigilance of EMA. And he's the chief editor in Frontiers in Drug Safety and Regulation, and member of editorial boards of drug safety, biodrugs, and clinical drug investigation. And so he's going to present an Italian distributed multi-database healthcare network, the VALORE, or I suppose in Italian VALORE project. And welcome to this meeting, and the floor is yours.
Thank you very much.
Yes, it's just a little bit difficult to hear you. But... because it's a little bit splitted, and I don't know. What?
Not really. Could you speak a little bit to see? Very badly.
Okay. Let me try with this. Do you hear me better?
Not really, but
it's not better.
Do you have a casque, something, or
do you have a problem with the microphone, or
do you have a headphone? Headset.
I'm not. Let me try with the headphone.
Qu'est-ce qu'on fait si ça marche pas? On passe au suivant.
Okay.
Ah, c'est qui peut. Leur téléphone, je sais pas. Comment?
Better?
Could you speak a little bit?
Can you hear me now?
Yes,
yes, it's better. It's better.
Okay, great.
Loud and clear, I suppose.
Okay, great.
Yes, it's perfect now.
Okay, thanks a lot for the kind invitation. Apologize for these technical problems. Very happy to present the Italian experience. So I'll be talking about VALORE project, but showing what we've been doing in the context of VALORE project. The aim is to show you what's going on in Italy with respect to post-marketing surveillance of drugs. So this is my disclosure of interest, as you can see. So please, next slide. The topic of my talk are summarized in this slide. So I will give you, first of all, a little bit of the background about where does Italy stand in terms of large-scale post-marketing surveillance of drugs, with some historical consideration. And then I will be talking more in depth about what we have set up in the context of post-marketing surveillance of biologics specifically, sharing with you some information about the approach that we have been using to conduct observational studies by employing a distributed database research network, with some also preliminary results. So next slide, please. So first of all, let me say that we talk more and more about real-world evidence. This is not at all an emerging topic. And in Italy, we have a very long tradition in terms of using healthcare databases for supporting regulatory decision-making. And you can see here in this slide that this is one of the very first database studies that was conducted in Italy to explore the association of gangliosides that at that time, we are talking about in the 90s, were among the top 20 drugs sold in Italy for cerebrovascular prevention mostly. And what has been done in this study, it was conducted as case control studies in some provinces of Lazio region, and the researchers involved the hospitals falling in those provinces, and they identified all the cases of Guillain-Barré syndrome. They selected controls within the same hospitals, and then they link the data of the information of these cases and controls to the electronic prescriptions of people residing in that area. So next. So in the end, what they've done using those prescription data, you can see the data date back at 1989. They've been able to identify next a 9 times increased risk of Guillain-Barré. Next, if you can go ahead. Okay, almost 10 times higher odds ratio. You can see here. And so showing a very strong association between Guillain-Barré and exposure to gangliosides. And thanks to this study, these drugs that were very frequently used at the time in Italy were withdrawn from the market. So here we are at the beginning of the 90s. So already we were supporting with the real world evidence regulatory decision making with even very extreme action like withdrawal of those drugs from the market. So next again. Okay, Italy is really rich country, both in terms of data. We have a lot of regional claims data, primary care, electronic medical records from GPs and family pediatricians, drug registries, and also very rich in expertise. There is a large Italian community adhering to the European Network for Centers for Pharmacovigilance and Pharmacovigilance. I have the pleasure to be part of the steering group since several years with other Italian colleagues as well. And if you can go ahead, please, next. And the participants, the Italian participants of this network, we all together, we summarized in this paper what has been done in Italy using healthcare databases for post-marketing surveillance of drugs with respect to both effectiveness, safety, but also pharmacoeconomic evaluation. So it's a very large community with very heterogeneous expertise. Next, please. And a few years ago, 2019 exactly, we organized a workshop in the context of the Italian Drug Agency to discuss about models that could be applied in Italy for establishing a database network, a research network supporting the regulatory decision making. And we invited key colleagues, colleagues from key experiences all over the world, from Sentinel, from CNODS, and if you can go ahead, next. And what we concluded is that, of course, we had the great potential in terms of data and expertise. What we still miss is a very strong governance of a model that may give the researcher the possibility to support systematically the regulatory agency. We are discussing in this recent time about on how to really consolidate those networks. Meanwhile, we are running several projects in collaboration with the Italian Drug Agency and the Italian National Health Institute of Health, mostly in the context of COVID-19 and vaccines. Several models can be applied, converting all the data in regional data in common data model, or developing ad hoc specific common data model. I'll be talking a bit later on about this issue. Next, please. Okay, why the interest on biologics? Next. As you know, a large part of drugs that have been marketed in the recent years, and they will be increasingly marketed in the next years, are biologic drugs, so drugs based thanks to biotechnology. And you can see in 2020, despite the COVID-19, almost 40% of the marketed drugs that have been approved by FDA were biologics. And also, we have also the monoclonal antibodies against spike belonging to this type of drugs. Very costly and also potentially affected by specific safety issues or deserving really specific surveillance system. Next, please. And I think everybody knows, is familiar with the difference between biotechnology drugs and small molecules in terms of complexity and also the dimension, the size of those drugs. Again, emphasizing the need to set up specific surveillance system. Next. Here is the examples of some safety issues that are specific for biologics, for instance, administration site conditions, infections, immune system disorder, and neoplasm. And you can see that in general, the safety profile of small molecules is completely different. Next. Next. And another issue is the marketing of biosimilars. So we have several biosimilars on the market. You all know about issues raised by the marketing of biosimilars, and especially the need to generate real-world evidence to increase confidence of patients and prescribers are using those biotechnology drugs. Next. And for this reason, several years ago, the Italian Ministry of Health funded a project to investigate the pattern of use of biosimilars and originator of epoetins and other first-generation biosimilars in several through a network of databases from several regions, exactly from 6 regions and some local health units. But the model we used was really rudimental, so it was really sharing raw data, of course, anonymized data, but very time-consuming and challenging. You can go ahead. Next, please. But still, we have been able to cover a population almost 15 million Italian citizens, almost 1/4 of the Italian population. You can go next. And these allowed us to investigate around 80,000 epoetin users and other biologics having biosimilars on the market. Next. 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 So these are the other drugs. And also we made sure we had the key information to investigate the biologics. So those drugs could be traced even if dispensed by the hospital. Biosimilars could be distinguished thanks to a drug-specific identification number. And also with the claims data we had access to, we could retrieve the key information for investigating those biologics in a real-world setting. Next. You can go. Please go ahead. Next. So these are all the papers that have been generated by this network. All sort of studies on biosimilars and originators, especially on switching between biosimilars and originator. Next. And all of this information supported both regulatory agency, Italian national agency, and regions that say manage the drug expenditure. And we developed also some innovative representation of one of the key issues, that is the interchangeability of reference product and biosimilars. You can see here, this is for the epoetins. So showing really the switching pattern among different originator and biosimilars, showing that around 20% of epoetin users switched at least once during the year. So the issue of switching and non-interchangeability is not only for biosimilars, but for the epoetins in general. Next. And also we performed a core study showing that whatever is the switch from the epoetin alpha originator, so if the switch is towards biosimilar or to other epoetins, there are no issues in terms of effectiveness and safety. Next. So this study was very important because thanks to the evidence generated by this study, there was a recommendation from ASCO clinical practice guidelines on management of cancer-associated anemia with erythropoiesis stimulating agents that support the interchangeability of different types of epoetins. So epoetins alpha originator and biosimilars, but also DARWIN epoetins and the other types of epoetins. So, but this is a very good example of how the evidence generated through these database studies in Italian database network studies inform also clinical decision making, because I mean this is a very important guideline from ASCO and ASH scientific societies. Next. But we have clear in mind that the most important biologics at the moment are the ones marketed for treatment of cancer, and they were marketed for treatment of autoimmune disease. You can see here that around 70% of the drugs marketed, biotechnology drugs marketed in the recent years, actually are drugs with approved indication for oncology and autoimmune diseases. Next. So for this reason, we had the possibility to have a very large-scale study funded by the Italian Drug Agency, VALORE project, that the project I'm talking about. We have several activities, several active surveillance studies in gastroenterology, dermatology, and rheumatology and non-rheumatology setting. But also the main core of the study of this project is the building of a large network of claims databases. Next. And the aim is to generate real-world evidence on the biologics that are used in those therapeutic areas, that to be used as source of information for healthcare professionals. In particular, we are carrying out a number of studies to investigate the prescribing pattern, but also comparative effectiveness and safety, also in relation to COVID-19. Thanks to the integration of claims databases and some clinical registries. Next. And you can see here, it was a kind of scaling up the previous study that we carried out on epoetins. Here we involved almost the entire nation, 54 million population out of 60 Italian population. Almost all the regions participated, and 16 out of 21, with some smaller one not participating. Next. And also on top of this network of claims data, we have the possibility to link the data. We are still exploring these with some clinical registries in the area of gastroenterology, dermatology, rheumatology from different regions. Next. And also with COVID-19 registry that was set up straight away after the pandemic beginning. Next. So overall, we can identify more than 160,000 users of biologics in the therapeutic areas I mentioned before, with also a large number of biosimilars. Next. What kind of approach we have been using for those studies? Next. We are developing a study-specific common data model. So we ask the different regions to convert their claims data into a study-specific common data model, and then we use a R-based tool called the Shinies to run locally analysis and generate anonymized analytical data set to be shared centrally for the central analysis, and all in a remote research environment, uplink in a remote research environment. Next. So we start first with the cohort identification based on the research question. For instance, users of biologics in those therapeutic areas, if you are interested in prescribing pattern of those drugs. Next. And yeah, based on the research question, we define a specific cohort, and then next. And then we asked each region to convert claims data into this common data model that is specific for the research question. Next. And we asked the region to install locally this R-based tool called the Shinies, where they have to there is input tables to be uploaded, and there is then a number of quality checks that this tool performs automatically. Next, you can see here the Shinies is being installed in different regions and is run locally by each region on the common data model, on the study-specific common data model. And this tool allows for quality check, record linkage of different data tables, aggregation, filtering, recording, covariate definitions, and in the end, we generate anonymized analytical dataset that can be integrated centrally and analyzed centrally. Next. And you can better see here that once the analytical dataset is generated, is uploaded in this CyberDAC. The dataset can be used by the coordinating center for the analysis, but also is uploaded in a dashboard that is present in this cloud storage, where each region can perform our analysis. So we facilitate also the analysis at a local regional level of the data on biologics. So it's very much appreciated by each region. Next. And several study design has been implemented in the Shiny. So there are several scripts that can be run based on the needs. Next. And as an example, here is a study that was conducted at the beginning of the pandemic on the ACE inhibitors and sartans. And you remember there was a concern about the association with the increased risk of COVID-19, as those drugs may increase the ACE2, that may be the entrance of the virus into our cells. In 50 days, something like 50 days, we've been able to adapt the Shinies and to run the study in this distributed database network. And from the ideation of the study till the submission of the study to the journal, we spent around 1.5 months. So it's really working with almost 50,000 COVID-19 hospitalized patients. So that was really a very good demonstration of the potential of this approach. Next, please. Next. And we are also using the same tool and the same approach in the context of different studies. For instance, on vaccines at the moment, we are investigating in association with myocarditis, with anaphylactic shock, and a number of other safety outcomes through the integration of data from different regions using the same approach. Some preliminary results from VALORE. Next, please. So the potential of this network has been described in this recent publication. Next. As I mentioned, we performed first a number of quality checks, and in particular for drugs like biologics, this is really needed because we encountered a lot of technical issues that may vary also across different regions. So quality check in those type of study is really essential, and it's really to be done as a study-specific approach. You can see here a nicely shaped increased prevalence of use of those biologics across regions. In a way, it's a kind of indirect validation of the quality of this of the data analysis. Next. And here we just computed the total amount of users in a 10 years study period in all the regions. So you can see here the contribution of different regions. I don't know if you can see properly, but to give an idea, we collected information on almost 60,000 users of adalimumab. And you can see on the right side over the total person years of exposure, Overall, next. Overall, we have more than almost more than 500,000 person years of exposure to biologics that are approved for the treatment of autoimmune diseases, and more than 35,000 users of biosimilars. So it's as interchangeability of biosimilars and originator is really something that has to be addressed in post-marketing setting, and with such a large amount of biosimilar users, we are in a position to explore more in-depth interchangeability for those biologics, interchangeability of biosimilars and originator. And also we can perform sub-analysis in very important subgroups that are not generally included in clinical trials, like children, very old patients, and pregnant women, and so on. Next. Also, we computed we calculated the sample power based on the number of users and the person years of exposure we collected. And here we simulated different incidence rate ratio to be investigated. Of course, the higher is the incidence rate ratio, so the stronger is the association we want to investigate, the lower is the need to accumulate person years of exposure. So you can see that the purple line is the one concerning a very strong association, but even for weak association like 1.5, so just 50% increased risk, there is the orange one. You can see that we can explore easily for almost all the biologics under study this association thanks to the collected person years of exposure. You can see, just to give you an example, for COVID-19, we estimated for investigating 1.5 incidence rate ratio for any of the biologics as compared to non-users of those biologics would be enough around 2,500 person years of exposure. And we have this exposure for most of the biologics. Next. And also, as I mentioned, we had a very large number of biosimilar users, and more than 15,000 were switcher. So really, we have great potential for investigating interchangeability. Next. And interchangeability not only between originator and biosimilar, but also between different biosimilars of the same compound. Next. So this was just an overview of what we've been doing in the context of this project. We are now extending the analysis to other regions and to data till 2021. So even increasing the potential of this network. And we are doing this in collaboration with Italian Drug Agency, setting up priorities. So this is a very good example of how the Italian community can liaise with the Italian Drug Agency. So to conclude, next. Well, this distributed database network has been setting up in several parts of the world, starting with Sentinel and other initiatives in Europe and also outside Europe, and in Italy as well. It's definitely a great opportunity for post-market surveillance of medicines and vaccines and biologics also, including biosimilars. Next. It's very important to have a number of quality checks, and also it's very important to involve in those quality checks the data provider, because they are the ones knowing the best the data, and there are a lot of technical issues that may have impact on the results and the findings later on. So we are really investing a lot of energy and time on this quality check. Next. And that's why it's very important to perform first drug utilization study, explore the treatment pattern of the drugs and of course biologics specifically. Also in relation to different local regional interventions. For instance, there were some interventions promoting biosimilar use in some regions that were more effective than as was done in other regions. Next. And we think that instead of converting a priori into a predefined common data model, the world database as done in other contexts like Sentinel, but also Odyssey, we think that having a study-specific common data model development may maximize the informativeness of the data sources. And these, of course, all true for biologics, but also for other medicines and vaccines. Next. And it's very important to set up those sustainable data infrastructure, because these can rapidly generate evidence supporting regulatory decision making, as was also highlighted by Anton in the previous presentation. And as mentioned earlier, in Italy, we have a great potential in terms of richness of the data and expertise. Next. So this was a very large consortium with a really very large number of very valuable people that made possible to generate all this information. And here you can see the ones playing a key role, especially colleagues from the Italian National Health Institute that were key in developing this tool called the Shinies, that at the moment has been used in almost 10 different large-scale post-marketing studies in Italy. Next. So these are the pictures of some of them, just to give them a tribute. Next, I think that's it. Thanks a lot. Sorry for the technical issue with connection, and I hope this was of interest for you, and happy to take any questions.
Thank you very much, Professor Truffaut. Don't we have some question? But waiting the question, I will ask you, because the VALORE project is a very mature network organization, and you were talking about stronger alliance needed between the Italian agency and research institution. How do you think that this can today be further expanded and even farther in a multinational perspective? You are mute. You are mute.
Yeah.
Thank
Sorry.
you.
So yes, I think the network can be expanded both nationally and internationally. Actually, we take part to several international networks. We start not all the regions, but most of the regions participating in VALORE project. They already participate in several large international networks, starting from already 2008. With a very important project called EU-ADR, that in Europe was the first to try to establish international database networks, developing tools like something similar to what we have done with the Shinies. At national level, what we are doing, we are investing a lot also in training, because what we want is not just to create a club of the best regions. In Italy, there are really huge heterogeneity in terms of expertise and capabilities and informatics skills of the people working at different regions, but we really want to involve all of them and support the ones really requiring also training in order to have a really nationwide studies. We are working on this on the Italian Drug Agency. At the moment, the collaboration is really project-based. That is, projects are funded to address specific research questions, but we are discussing with the governance of the Italian Drug Agency to set up a model similar to what has been done, for instance, with Sentinel, or similarly to what has been done at EMA also level with the DARWIN Coordination Center. With respect to that, I think DARWIN may also help in facilitating international collaboration. So I think it's really very dynamic environment, and I think there will be a push towards establishing international networks. Also, I mean, based on what DARWIN will be doing in the next period.
Thank you. Vincent, we have other question on the chat?
Yes, we have a question. Indeed, your presentation provided a clear example that connecting data allows reaching adequate number of data to allow scientific analysis. But about the linkage with other claims data and common data model generation, could you have example, what were the difficulties you were facing, and if any, how did you solve it? for example, about quality check or whatever.
It depends on the additional data that will be linked to those network of claims data. For instance, we have done several studies linking claims data with the COVID-19 registry. That was actually quite easy because the way this registry is really regional based and is treated in a similar way to how it's treated, how the claims data at regional level are treated. So basically, we have a anonymized patient ID, and we can easily link claims data with COVID-19 registry at regional level. So that was quite easy from a technical perspective. Also, no issues with data privacy. Sometimes some regions there are data privacy officers that are more critical than others. So this is, I think, a general issue. But yeah, it depends on the type of data. Of course, again, quality check is very important. It's very important, again, to involve people that have a key role in the generation of those data, especially with respect to clinical registries, because there are issues that may be encountered that they need quickly an interpretation, and for that it's very important to involve data providers.
Thank you.
Thank you very much. So it's okay. No more question. I think it's time to have a break, a coffee break in Italy, a tea break in other part, and I invite you to come back at 3:40 sharp. And thank you, Professor Truffaut.
Thank you very much.
Welcome back to the RWE conference. We are going now to pursue the session by hearing Edward Byrne on generating evidence using an international common data model. Edward Byrne is a postdoctoral researcher at IJAB Jordi in Catalonia, Spain. He has experience using common data models to perform research, and in the last couple of years has applied this to the topic of COVID-19. This was first to help understand the profiles of patients with COVID-19 and their outcomes, and laterally for assessments of the safety of vaccines against COVID-19. Please, Edouard, welcome, and the floor is yours.
Thank you. Thank you for the invitation to speak today. Is my sound quality okay?
Yep.
Yeah,
it's
okay.
Perfect.
So I'm just waiting for the presentation to load. Okay, here we go. Perfect, thank you. So yes, I'm here today to speak about our work using common data models and specifically the OMOP CDM. So what I'll do is I'll give a brief overview of the OMOP common data model, after which I'll talk through some case studies where we've used it in recent times. So next slide, please. So this image is to give an idea of the typical approach to research using patient-level data. So it's this often quite time-consuming process, taking data that's perhaps not collected for the purposes of research and just transforming it into a structure where it's ready to be used, and then applying certain analytic methods to generate hopefully reliable evidence. Next slide, please. The idea with the common data model is to add this intermediate point where this data preparation stage is standardized between databases, and all databases are standardized to the same common data model, after which analytics can be used. So the key point I think here is that next slide, please. Sorry. Next slide. So this is the OMOP CDM, and I think the key point here is that this is a data model that's not study specific. This is a model that's generic, and so patient level data in a database is mapped to this common data model, and then after which it is used for various research questions. Next slide, please. So this is what the data model currently looks like, with each of these boxes being a table in the database. Next slide, please. So to start with, on the left-hand side, we have all the tables containing patient-level data. So this is a person-centric model where we have this person table which contains the person IDs, the unique identifiers for all the individuals, and some demographic information such as date of birth. And then there's various tables that can be linked to this person table. So for example, we have a condition occurrence table, which contains the various diagnoses a patient might have recorded. And here, importantly, these diagnoses not only have the date, but they have a standard concept associated with them. So in terms of the condition occurrence table, SNOMED codes are used to represent conditions across databases. So this clearly helps to facilitate network research by using the same vocabulary. Next slide, please. And then the other kind of set of tables I would just like to mention are these vocabulary tables. So these are what facilitate the mapping between source data to the common data model. So for example, if a source database is using ICD-10 codes to represent conditions, these vocabulary tables help to map these ICD-10 codes to SNOMED codes and so on. Next slide, please. So the first part of the process is getting to the point where the patient level data is in the common data model. Next slide, please. And here, because we're using an established common data model that's used by many different research groups, there's many tools that have been developed that can facilitate this mapping of the database, of a source database. So for example, for designing the ETL, we have tools such as White Rabbit to profile the source data. rabbit in a hat to describe the mappings of the source data to the common data model. We then have tools that aid the implementation of the ETL. So, for example, Usagi, which would provide suggesting mappings from source codes to standard codes. There's then tools to help create the database structure. And then importantly, there's tools to assess the quality of the data once mapped. So the one shown here is Achilles, and this will provide an overview of the data that's been mapped to the common data model. So this isn't about study-specific data quality, but this is just about the data quality of the map database as a whole. Next slide, please. And recently, this has been added to with another tool called the Data Quality Dashboard tool. And what this does is it runs thousands of data quality checks on data that's mapped to the common data model. First, to make sure that the database conforms to the structure of the model. Second, to ensure that the required fields have been populated. And third, to make sure that the values that have been inserted are plausible. And this helps to provide a level of trustworthiness to the map database. Next slide, please. So that's the path of getting to the common data model. Once we have a database mapped to the common data model, we then have a number of ways we can go about analysing the data that's been mapped. So next slide, please. So the first option is to write custom code to query the database and to produce evidence. So, for example, if this is a new bespoke method that's not been used before, you might be in a situation where you need to write code to generate evidence. However, there's also existing open source methods, library packages that can be used to run standardized analytics. So, for example, there's packages to run propensity score matching and so on. So where we have studies that are similar to those that have done before, often there will be existing tools and code base that can be used. And the last option is there's a graphical user interface that helps to run some standard analytics, so basic descriptive statistics in general, and it also can help with generating phenotypes that can be used to create cohorts across different databases. And all of this can be used to generate this reliable evidence that we're after. And depending on which of these options is required, that will obviously have an implication for how much time each analysis would take. Next slide, please. So that's the very kind of brief, high-level overview of the common data model. I think probably what would help is to talk a little bit about the studies that we've performed, which would hopefully give a clearer idea of what this looks like in practice. So the first case study is about understanding COVID-19 patient profiles. Next slide, please. Next slide, please. So here I'll talk about two studies we've performed, both of which were done in 2020. Next slide, please. The first of which was a single database study. However, that database was mapped to the common data model, and that study was looking at the natural history of COVID-19 during the first wave in Catalonia. So next slide, please. So the objective of this study was first to summarise COVID-19 outcomes in Catalonia, and then the second objective was to describe the associations between age, sex, and comorbidities and the risk of various COVID-19 outcomes. So being diagnosed with COVID-19, being hospitalised with it, or having a COVID-19 related death. Next slide, please. So this study was informed with data from CIDIAP. So this is a database which captures primary care records from approximately 80% of the Catalan population. This was also linked to hospital data on a patient level basis. This database was an existing mapping of the database to the OMOP CDM already existed prior to COVID, but with certain new diagnostic codes and so on, this was updated to incorporate the COVID data as it came on board. But the end analysis was run using the OMOP Common Data Model. The study period began on the 1st of March 2020 and went up to the 6th of May 2020, which was at that point the latest data we had available. We used a multi-state cohort model as a framework for analysis, with all individuals beginning in a general population state. Next slide, please. So hopefully this slide shows the study design quite clearly. So we've got this general population cohort, which essentially is everybody in CIDIAP as of the start of March 2020 without a history of COVID. So this is where the study starts. And then the use of a multi-state model allows us to look at the trajectories of patients with COVID-19. So we can see 100,000 people that had a primary care diagnosis of COVID-19, of whom over 8,000 went on to be hospitalised. There was also 8,000 people that were first seen being hospitalised with COVID-19. That is, we didn't see an outpatient diagnosis of COVID-19 prior to their hospitalisation. And then we have the deaths after diagnosis of COVID-19 or after hospitalisation with COVID-19, but also among our general population cohort, with this study going up to May, as I said before. Next slide, please. So this first let us look at the age profiles of the people making the different trajectories. So we have our general population cohort, which is everybody in CIDIAP as of March 2020, and we can see kind of the age distribution there. We can see that those diagnosed with COVID were older than the general population, and then those being hospitalised were older yet again, and those that died after hospitalisation were older than those hospitalised in general. So again, this is quite a simple analysis, but it hopefully highlights what can be generated using the data in the Common Data Model. The next slide. Here we look at the relationship between sex and the risks of the different transitions in the model. So the left-hand column is the overall association, and then we've got different stratifications, so by month, by age, and by region within Catalonia. But essentially what we see is that being male was associated with a lower risk of being diagnosed with COVID-19 as an outpatient, but after which all other transitions, males had a higher risk. So once diagnosed, being male was associated with an increased risk of being hospitalised, and once hospitalised, being male was associated with an increased risk of death. Next slide, please. Here is a very busy figure, but this is where we looked at various comorbidities and their association with the different outcomes of the study. So we've got both overall and then by age and sex strata. So I won't go through every single one of these, but this is just to show that we were able to look at the associations between all these different conditions and outcomes. So for example, we could take something like type 2 diabetes, and we can see that the increased risk was most pronounced for females who were under 70. And next slide, please. Yes, so that was a study that was based on a single database that was mapped to the OMOP Common Data Model. This second study is a network study. So here we brought together data from multiple databases from across the world. This was a study we started towards the end of March 2020, so right at the beginning of the pandemic. And as data became available in the different databases, this was added to the study. So I remember the first draft of this included 1,000 patients, and within a few weeks, while we were waiting to revise the paper, we got up to 30,000. And I suppose if we'd waited another 6 months, it would be many more times that. But next slide, please. But here the study was to describe the characteristics of patients hospitalized with COVID-19. And then our second objective was to compare the characteristics of individuals hospitalized with COVID-19 to those of patients hospitalized with influenza in previous years. Next slide, please. So as well as using data from CIDIAP in Spain, we used data from South Korea, the US, and also another hospital database from Spain. All of these databases were mapped to the OMOP Common Data Model, and we summarized the demographics, the conditions, and prior medication use of those being hospitalized. Next slide, please. So in total, we included over 34,000 individuals hospitalized with COVID-19, and different databases contributed different amounts, with CIDIAP contributing 16,000 patients hospitalized with COVID-19 in Catalonia. Next slide, please. So this slide shows the prevalence of all the different conditions and prior medication use that we looked at in each of the databases. And I think this is quite a nice demonstration of the richness of data that we have once mapped to the Common Data Model. So we were able to look at thousands of different conditions and medication use and plot their prevalence here. Next slide, please. As well as this, we were able to look at the comparison between those people being hospitalized with COVID-19 and those people being hospitalized with influenza in previous years. So we have this diagonal line, and on the right-hand side indicates that the condition or the medication was more prevalent among those being hospitalized with influenza. So we can see that aside from being male, pretty much everything else was more common among those with influenza. And what we can take from that is the people typically being hospitalized with influenza generally had more comorbidities and prior medication use than those we were seeing being hospitalized with COVID-19 at that time. Next slide, please. So that was the very initial work we did on COVID-19 during 2020. The second case study is much more recent, and that's been looking at the background rates to help assess COVID-19 vaccine safety. So next slide, please. So this is very recently published in the last few weeks, and this analysis was looking at background rates of different thrombosis with thrombocytopenia syndromes, which have become of special interest for COVID-19 vaccine safety surveillance. Next slide, please. So here our objectives were first to estimate the background incidence rates of non-vaccine induced thrombosis with thrombocytopenia. So thrombosis with thrombocytopenia we see in earlier years. And our second objective was to describe the profiles of individuals who have non-vaccine induced thrombosis with thrombocytopenia. Next slide, please. So for this study, we used data from seven databases from Europe, so from France, from Netherlands, from Italy, Germany, Spain, and the United Kingdom. We included individuals who were present in the database as of the 1st of January 2017, and we followed these individuals up until the 31st of December 2019, which was the end of the study period. In the analysis, we calculated the incidence rate per 100,000 person years for the various events of interest. Next slide, please. So one important consideration here is the differences between the databases. So I think this is useful to note before seeing the results. So although all of these databases include primary care data, they vary in hospital linkage, with only 2 of the databases having patient level linkage to hospital records, with one being CIDIAP and the other being HIC Dundee from Scotland. They all had outpatient platelet measurements, which is obviously important to identify thrombocytopenia, but only one of the databases also had inpatient platelet measurements captured within their database, which was HIC Dundee. Next slide, please. So we included over 38 million individuals in the study, with more than 13 million from CPR-Diorum down to a little over 1 million for LPD Italy. Next slide, please. So this figure, which maybe is a little hard to see, is showing the incidence rates for thrombotic events of interest. So at the moment, this is without the combination with thrombocytopenia, but thrombotic events. So the top one would be CVST. And then going down, we've got the more common DVT and pulmonary embolism. And we can see the kind of age relationship, which is well known for these events. But we can also see quite substantial heterogeneity in results across databases. So just picking out pulmonary embolism, for example, we can see that the highest incidence rate was for HIC Dundee, which to go back was the database which did have hospital linkage. Next slide, please. So this is plots of thrombotic events with thrombocytopenia. So these were far rarer than thrombosis themselves. But again, there was heterogeneity between databases, and here even more so really, which underlined the importance of the degree to which platelet measurements were captured. So HIC, which had both outpatient and inpatient platelet measurements, had a much higher incidence rate than other databases which only captured outpatient measurements. So this wouldn't necessarily be unexpected, but I think underlines the importance of understanding where the data is coming from when interpreting the results. Next slide, please. And here we have the characteristics of the study populations as a whole, so in grey bars, and then for each colour is the populations of persons that had an event of interest. So here we can see those people with an event of interest much more frequently had a comorbidity or medication use of interest prior to this event. Next slide, please. So yeah, that's a few examples of the studies I've been involved with that have used the OMOP-CDM. Next slide, please. So I think what we can see is that standardizing data to this specific common data model has facilitated both local and network studies on COVID-19. What it's meant is that having the existing mappings in place meant that we could leverage existing open source tools both to map data, the original data, and also new data that came on board during the COVID-19 pandemic, and also to facilitate rapid analysis while maintaining quality throughout. However, it should be noted that there's heterogeneity between databases, and this can be expected. We see that even once we standardized to a common data model, run the same code across databases, we still see heterogeneity in results. Probably thrombosis with thrombocytopenia is probably an extreme example because of the different domains identifying it requires, but that's certainly going to be seen in other studies as well. And I think that's the end of my presentation.
Thank you, Edouard. Thank you very much. Thank you for this presentation. We have time for questions now. I am talking to the audience. Do not hesitate to write questions in the chat. During the presentation, there is no problem. We are taking it at the end.
In language.
And first of the question, maybe to complete your presentation, you were talking about the use of data from South Korea and FDA. I am aware that you are setting up a platform and setting up collaboration with South Korea and FDA. Could you tell us a little more about this platform?
Yeah, so I hear a bit of echo. Maybe
Yeah, we can hear you.
Maybe that's better.
Yeah.
You hear me okay?
Yeah, it's okay.
Yeah. So I'm a collaborator in what's called the Odyssey Network. So that's an open science community that supports the use of the OMOP Common Data Model, and that has partners from across the world, really. So there's many partners in the US, and there's also partners in South Korea and across Europe now. And yes, they've all mapped their data to the OMOP Common Data Model. So it's about applying standardized analytics to this map data. More recently, I've also been involved in European projects such as Eden, and will be involved in DARWIN, which are also built around the OMOP Common Data Model.
Thanks, Theo. So we will be talking about DARWIN in a few minutes, but the second question, maybe, we were talking during the other presentations about collaboration between academics and regulatory. In your works, did you experience collaboration with the regulatory agency, and how was it?
Yeah, so that last study was funded by the MA, in fact, and I think, well, I think it was successful, and I think one of the things that made it successful is this focus on data quality, both study specific but also at the database level. So often it's difficult to ascertain the quality of the data that's being offered for studies. However, the increasing use of these tools, such as data quality dashboard, really can help provide some confidence that the data is research ready, and also looking in detail at the impact of different inclusion criteria, different ways of defining study populations, and their impact on the results. I think was an important contribution to the success of the project.
Thank you. Maybe a point about the skills that are necessary to develop common data model, as you were presenting us. Indeed, you are working in a team, obviously. Could you tell us about the skills that are critically needed, and maybe the new skills that are needed and maybe developed in your team?
Yeah, so mapping data to the data model is a huge undertaking, and it's not a one-off undertaking. It's something that needs to be repeated over time as new data arrives, whether that's an update with the same structure as your existing data, but just with more follow-up time. But also often you'll get new variables. And then on the analytics, there's plenty of complications there too. I think the key skill here is collaboration. So previously, many studies have been done kind of independently by a small number of researchers, and there's an expectation that that small group can do every step of the process. Whereas with these large projects, it requires expertise across quite a wide range of topics. So really it's about bringing together people from different research institutes, bringing in clinical knowledge as well as data science knowledge. And yes, I think that's probably the biggest thing is getting people to work together. And one of the nice things I think with working with the OMOP Common Data Model and the researchers associated with it is the focus on open source. So all code is made publicly available from a study after we publish it. So that means it can be used and improved in future studies. And I think that's been a really beneficial point, both in performing specific studies and developing the infrastructure over time.
Thank you very much. And you are, I think, an example of collaboration because you are in this as a postdoctoral position, and you are from UK and working in Spain and part of Catalonia, more specifically, and you are evolving in a multinational team. Indeed.
Yes. So I think that's one point as well is the movement of individuals between research centres to kind of share best practices and learn from others. So when we're running a network study, you really need to understand each of the centres, each of the databases contributing to it. So I've been fortunate enough to yes, work in Catalonia. I'm also have an affiliation here in Oxford, so that's where we use the CPRD data. I've spent some time at IPSI with the data from the Netherlands, and I think, yeah, getting that knowledge of the different databases is key as an analyst in terms of understanding the output that you get once you run the study.
I think we have a question from the chat. Our colleagues are taking it, and in a few seconds I can read it. Do not hesitate to ask questions. We have a little time before the next presentation from Xavier Kurtz, and our team from ANSM is sending me the question. It is a long question. Very, very long question. I do not know if the answer will be as long as the question.
Or French questions that need to be translated in English, perhaps.
Yeah, do not hesitate. I am speaking to the audience to write your question in French or in English. We are translating it. So the question is, besides heterogeneity in background IR between different databases in different countries, we also observed, for example, for myocarditis, a wide range in background IRs, even within a single database, which makes the OE analysis challenging. Do you have any ideas how to deal with this heterogeneity?
Yes, so I think it's a difficult issue. So if we take a single database, for example, I think a key thing to note is the importance of phenotyping. So there's generally not one single code we're looking for. We're looking for some combination of codes that indicate a condition or an outcome of interest. And depending on which codes get included, that can have a very large effect on the estimate at the end of the process. So that's key in terms of understanding the impact of these algorithms to identify patients. So that's probably the first point. And I think the second point is it just perhaps underlines the importance of using comparable data sources when making comparisons against background rates. So if we have one database and we estimate that the risk of an event after vaccination, for example, or maybe it's coming from case reports, any comparison with another database from historical rates from another database should be treated with quite a bit of caution, I think, is probably a key takeaway.
And how did you define the more specifically comparable database? Do you have a more technical definition of comparable database as the biosimilar and the bioexact that we had in the last presentation?
Yeah, so I think we can think of databases in different ways, right? But for example, primary care databases is one group of databases. So we can limit an analysis to primary care data if we think that will give us kind of the population of interest. Perhaps mixing up a primary care database with a claims database from another country, it would be unsurprising that there would be heterogeneity in the results. Another point is what linkages are possible with the different databases. So we've seen the importance of whether there's been linkage to hospitalization records and the degree to which measurements are captured within the different databases. But I think this is also a strength of running network studies, in that if you run a study in a single database, you'll often end up with one number, and kind of your takeaway is that's the estimate that can be applied for the situation in any given context. Whereas running studies across a network of databases can show just how wide-ranging estimates can be, depending on where you're looking, which is not necessarily a bad thing to show, I think.
Thank you, Edouard. I think we can end this session, and thank you for answering the questions. I propose, I wonder if Xavier Kurtz is in the starting block for its presentation. Is it, if he's okay? Hello, Xavier. Okay, so I give the floor to Jean-Michel to introduce the next presentation, and thank you one more time to Edouard.
And welcome to this meeting. I remind that Xavier Kurtz is a medical doctor specialized in tropical medicine, and you have been working for several years in public health projects in Africa, Asia, and all over the world, I suppose. And you are also a PhD in epidemiology and biostatistics at McGill University in Montreal, in Canada. And you are working in the Department of Pharmacology of the University of Liège in Belgium, and in the Belgian Center for Pharmacovigilance at the Ministry of Health. And you currently lead the data analytic work stream at European Medicine Agency, and you are going to talk about enhancing use of real-world evidence for regulatory purpose, and about the DARWIN network and the EMA registry initiative. And I remind that it's already possible to ask some question in the chat in English or even in French, any language, not any language, but English or French. And Xavier, the ground is yours for your presentation.
Okay, thank you. My initial title was on the EMA registry initiative. However, given the fact that we have launched DARWIN EU framework just recently, a few days ago, I would say, it was agreed with the organizer to have this topic in my presentation. So I do not have my slides, if I can have them. Okay, so thank you. So in my presentation now, I will first explain the use of real-world evidence in regulatory decision-making. I will then present the DARWIN EU network, and then I will go back to the use of registries in marketing authorization applications and give a few words on the CHMP guideline on registry-based studies that was published at the end of last year. Next slide. So the first question is, how do we define real-world data and real-world evidence? We define real-world data as routinely collected data relating to patient health status or the delivery of healthcare from a variety of sources other than clinical trials. And there is a very large variety of data sources. And currently, the ones that we are using, that we are accessing more frequently, are those containing data on electronic healthcare records from ambulatory care or specialist care, hospital data, registries, claims data, or mobile health. But the situation is progressively changing, because now we have access, we are starting to have access to much rather broader scope of data, like genetic data, which may be available from some data banks. Real-world evidence is defined as information derived from the analysis of real-world data, but it should be stressed that this is not any information. This is high quality information that is relevant, valid, reliable, and appropriate for regulatory decision making. Next slide. There are three main areas of committees decision making for which real-world evidence can be requested to marketing authorization applications. The first one is to support the planning and validity of applicant studies, to understand the clinical context, and to investigate association and impact. And I will detail a bit these three domains of real-world evidence for regulatory use. Next slide. Let's start with the support to the planning and validity of applicant studies. And we have here identified two main case categories: the design and feasibility of planned studies, when there is a plan to conduct studies to inform the recruitment in pre and post-authorization studies, including the number of incidents and or prevalent patients per year, to examine the impact of the planned inclusion or exclusion criteria on patient recruitment and the feasibility of the study, and also to look at the composition of the study population versus real-world target population. And then the representativeness and validity of completed studies can be evaluated, and this will be done by evaluated external validity of completed studies by measuring the representativeness of the clinical trial population versus a real-world target population, also to evaluating whether the standard of care used in the control arm of clinical trials is comparable to the current real-world standard of care. Next slide. The second domain that we have identified is to understand the clinical context for applications. And we have here three main use categories: the disease epidemiology, clinical management, and drug utilization. Disease epidemiology is to support the evaluation of the incidence and prevalence of disease, and therefore support better understanding of the disease and its progression over time. For the clinical management, this may generate evidence on the actual clinical standard of care and the comparison of different populations in order to know how patients are currently diagnosed and treated, and therefore to assess what could be the added value of new medications. And then for drug utilization, this allows to characterize real-world drug use in terms of incidence, prevalence of use, indication, amount and duration of exposure, and switching of drug use over time. Next slide. The third domain is to investigate association and impact. This is a bit more classical domain that is used more frequently now and over the last decades, but this is an important one. And there are two main use categories that we have identified: the effectiveness and safety studies, and then the impact of regulatory actions. Well, the effectiveness and safety studies have been done for a long time with real-world data, as was mentioned by Gianluca Trifigro, and this allowed to investigate associations between treatment exposure and outcomes, which may be effectiveness or safety outcomes. It also allows to characterize adverse events in the context of the pharmacovigilance evaluations. And then the impact of regulatory action is something that we have introduced a few years ago, where we really wanted to monitor the implementation and the effectiveness of risk minimization measures that were decided by regulatory authorities. So if there is a change in the summary of product characteristics or restrictions in the use of a product, do the decision led to any change in the treatment, and is there any evidence that the change, that change led to safer treatments? It's not so easy to assess this, to make this evaluation. However, methods are being developed now in order to have a better understanding of what is the effectiveness of the regulatory decisions, and how can we assess it, and how then can we improve these decisions. Next slide. So let's go to the DARWIN network, which will be one of the main. We hope that will be one of the main sources of information to perform this real-world evidence studies for regulatory purpose. So it has been launched end of February. This is a federated network of data, expertise, and services that supports better decision-making throughout the product lifecycle by generating reliable evidence from real-world healthcare data. So this is the vision that we have. And by federated network principles, we mean this is a network of data source where the data stay locally, and a common data model is used to perform studies in a timely manner and to increase the consistency of results. So I'm very glad that Edward Byrne has explained what is a common data model in the previous presentation, so I don't have to do it now. Who are the actors of DARWIN EU? DARWIN EU will be operated by a coordination center, and the coordination center will interact with different organizations. First of all, EMA, which will have an oversight on the activities of the center, and it will also interact with the scientific committees to identify research questions and provide results. The coordination center will also liaise with the European Health Data Space, so the EHDS network that is being started now, and it will interact with the data permit authorities that are planned to be established in member states to support the access to data source available. And the coordination center will, of course, then directly interact with the data partners that will be included in the federated network of data. Next slide. I said that DARWIN EU will provide data services and expertise, but what does it mean in practice? It will maintain a catalog of known relevant data holders, continually ensuring the discoverability and quality of data being held by the holders. It will maintain and expand then the federated network of data partners. So these are data holders that are conforming to the required standards for usage in the regulatory context, so that can show and that there will be process for that to assess the data quality that is can be available from these data sources. And then the coordination center will conduct scientific studies and analysis on behalf of the European Medicines Regulatory Network and also the EMA scientific committees. There will be training, governance, and also that the coordination center will allow the Medicines Regulatory Network, EMA, and scientific committees to make use of the EHDS system in the context of medicines regulation. Next slide. So I mentioned that DARWIN EU will provide data services and expertise, but what is it? Well, yeah, this is then in terms of conducting studies, what does it mean? And what will be the process? First of all, the questions will be identified or will be raised at the level of the national competent authorities or EMA committees. These questions that may help making decisions on authorization or pre or post authorization will be translated into research questions. There will be feasibility assessment, and then research question will be communicated, transmitted to the coordination center that will create study protocols and programming codes, and will then contact data partners. The data partners will receive and run the code on their own database. They will send aggregated data to the coordination center, and then the coordination center will compile this information and then communicate these results to the requester that will then be able to include this information into the decision-making process, into the regulatory procedures. Next slide. We have identified 4 main categories of studies, and this is important because these different categories will have different timelines. Some of them, the first categories can be performed very quickly, and also in terms of feasibility, they may be easier. First of all, the routine repeated analysis. They are based on a generic study protocol, and they consist in repetition of the same analysis for the same product. For example, a periodical estimation of drug utilization, safety monitoring, or estimation of the incidence of adverse events. So once we have programming codes, and this was, as Edward Byrne explained, so this is a series of code that can be reused and then just run at the level of the databases locally. Then what we call the off-the-shelf studies are the studies for which a generic protocol is adapted to a research question. This is still a rather simple design to estimate prevalence, incidence, or characteristics of exposure and describe population characteristics. Complex studies are those that perhaps are more the studies that are more traditional in terms of epidemiological terms. These are the studies that may assess associations, that may measure strengths and determinants of the association between an exposure and an outcome. So this may be more complex, require more expertise probably, and more complex data source probably. And so there is a more development and customization of a study protocol. And then we have the category of what we call very complex studies, which cannot rely only on electronic scale database, but which may require additional data collection, primary data collection, linking different type of databases. So this may be probably less frequent study. We consider that the routine repeated analysis, the coordination center will be able to do many of them, and dozens of them, I would say, in a year. Off-the-shelf studies, probably 2 complex studies and very complex studies, this will be less frequent. Next slide. Just in terms of timelines, so last year we selected the coordination center based on a competitive tender. We then appointed the coordination center in February, and this is the Erasmus University Medical Center Rotterdam, who is leading a consortium of centers. And so this year and next year, there will be what we call the establishment phase, where the coordination center will be set up. There will be connectivity with the European Health Data Space. There will be the first catalog of standard data analysis, and also recruitment of the data partners, ensuring that these are data partners that can provide high quality data and reproducible data. And we will then also start running pilot studies. Now, from 2024, the network should be operational and be able to routinely support scientific evaluation by European committees or others. And then in 2025-26, it will be fully operational. So we have currently a 5-year schedule for operations. Next slide. Who will benefit from Darwin? First of all, as I said already, EU medicines regulators, and I explained the use cases for drug development, authorization, and pre-authorization and post-authorization, but also it is intended that in the medium to long term, much more organization will benefit from Darwin EU. EU patients and healthcare professionals could have access to information on innovative medicines and their use, safe and effective use. European Commission, national computer authorities for different types of applications like health policy and delivery of healthcare, health technology assessment bodies and payers, health agencies, EU international, academic and research academia and research organization may have then access and may use then real-world evidence, and this could be a good platform providing high-quality data for methodological development, and also at the end, industry may benefit from better evidence supporting decision making. Next slide. So now I'm coming back to my initial topic, which was registries, and why did we have an interest in registries? This comes from a study we did several years ago on the number of registries imposed and an obligation to marketing authorization application at the time of authorization. And this was a period of 2005 to 2013. What we saw at that time is that the use of registry was imposed for 9% of the products that were authorized, but the large majority of these registries were product registries, and 80% were new registries. And then, so they were not, they were specifically built for that study, for that request. However, there were many problems, and you see that for more than 50% of the studies or registries, there was a low accrual rate. A large number of the registries required an amendment of the protocol. There were, for many of them, low data quality, low use of the product. So there was a decision that we should try to do better than this. And next slide. We then started in 2015 the EMA Patient Registry Initiatives, which is based on a cross-committee task force on registries, with the aim to facilitate the use of disease registries by introducing and supporting a systematic approach to the contribution to the benefit-risk evaluation. And there were two main components. First of all, to interact with regulators, companies and registry holders to understand the barriers, opportunities of using existing disease registries, and then to provide guidance on methodological concept and requirements. Next slide. So we also, in the context of this initiative, we organized a large number of multi-stakeholder workshops on cystic fibrosis, multiple sclerosis, CAR T-cell therapies, hemophilia, also cancer therapies based on tumors, genetic and molecular features. And all these workshops addressed issues of data elements to be selected, to be included, on governance of the registries, on the quality of the data and what are the minimum requirements in terms of quality, and also in terms of sharing of data. And then we hope that it has been successful, but I would like to show some results of a more recent study that we did, and starting next slide. Next slide, please. So we did 1.5 years ago, a new study which was not based specifically on registries, but on the use of real-world evidence in marketing authorization applications submitted in 2018, 2019. So this is the submissions, it's not only the authorizations. And we saw that for initial marketing authorization application, nearly 40% of the applications included some components of real-world evidence supportive or contributing to the authorization. For most of them, the data would have been collected post-authorization, and then also for extension of indications, this is 18% with data to be collected pre and post-authorization. One aspect that is important to say here is that although we had a definition of real-world data, it's very difficult to see what is the operational definitions when we did the study. What do we include as real-world data or what we do not include? And there have been several studies, more or less the same type of studies, finding very different results. And so one study that we have seen, one recent study that found nearly 100% of the authorization had real-world evidence components. Others found much less. So this is a bit comment here, but there is certainly a need for harmonization and of the definitions of real-world data, real-world evidence. Next slide. And we see that among the results, safety objectives were for those that were mostly used for use of real-world evidence, and then the cohort studies were the majority. So this is just a few of the results. But the next slide is most important. Actually, this is the one I wanted to show. Next slide, please. Is the fact that we saw that among all the applications, so from new market, again, this is 158, I think. 24% of the authorization had registries as a source of real-world evidence. And so this figure, 24%, is compared to 9% that we had several years ago. And also that they were representing 60% of the source of real-world evidence in the applications, and disease registries were the majority of the registries that were used. So I said this to show that actually in a matter of perhaps 5 years, existing disease registries are much more used and have as a source of evidence supporting marketing authorization application in the different use cases that I included, like being used as information to on the standard of care, on the disease epidemiology, on the comparator groups for single-arm trials or for comparing placebo groups. So it seems that using the existing disease registries as a source of data to support authorizations is becoming more and more important. And I think that this is a very, very nice progress in this field. Next slide. Just a few words on the CHMP guideline. This will not be very long, but this was published last year, and this takes several years to be finalized. And this is good that actually it comes really at the same time as use of disease registries is becoming is increasing, and as I show, is much more frequent now. The aim of this guideline is to provide methodological recommendations for studies that are being based on registries. So there are a number of recommendations on on the study protocol, study population, data collection, quality management, analysis, and reporting. There are also some information and some recommendations on the legal basis and regulatory requirements to clarify a number of aspects and answer questions that we had in terms of regulatory requirements for using registries. Next slide. Next one. Okay, the scope of the guideline is mainly studies based on disease registries or studies characterized by the presence of particular conditions like pregnancy registries or registries of birth defect. And we use the definition of a patient registry that is about similar to the one used by the FDA and the US Agency for Healthcare Research and Quality, that it is an organized system that collects uniform data to identify specified outcomes for a population defined by a particular disease, condition, or exposure. And then I would like also to highlight that we do not like anymore the term product registry, because according to the new regulation, new legislation, to the new clinical trial regulation, this term product registry fulfills the definition of a clinical study. So actually this is either a clinical trial or a non-interventional study. So a product registry is now a misnomer in terms of at least in regulatory terms. Next slide. Registry-based study is an investigation using the data collection infrastructure or patient population of registries. So this is rather straightforward. Next slide. I would like just to highlight one thing is that we, when I say we, it's the task force, has introduced the feasibility analysis that would like to be, that regulators would like the companies to perform with the registry holders before proposing or before really making studies based on registries. And this is to answer questions on the suitability of a registry for a specific registry-based study that is being requested. The question is can the registry really answer the question that that is being asked? And I have to say that often this is not always the case. And then it's better to have this answer early in the discussions and the authorization application procedure rather than too late. Next slide. One thing is that we have an annex on consideration on patient registries. The medicines regulators and especially EMA do not have an authority to provide recommendations to registry holders because registries need to follow the national legislations. However, it's important to mention some information that regulators think important to consider when using registries for studies. So these are desirable elements that registry holders should consider if they want for being used for registry-based studies, and the elements of data elements, quality, governance, and so on. Next slide. Just one thing, we also included some responsibilities for safety reporting, and this should not be read, but we had the EMA and with, I'm sure, in the level of the national competent authorities, a lot of questions on what are the obligations for safety reporting from registries, and we try to clarify that and at least to provide all the possibilities. And I then arrive to my conclusion. Next slide. That real-world data and real-world evidence are extensively used for the post-authorization safety monitoring of medicine and can support pre-authorization evaluation. So post-authorization safety monitoring, this is for a long time now, for decades that they have been used, but the use of real-world evidence to support pre-authorization evaluation is becoming more frequent now, and it's probably in the coming years it will be nearly a must or a common feature. DARWIN EU is being established to support the regulatory network for analysis and studies, but what is important to say is that the focus is really to collect data of high quality, and that's validated data that can provide valid and reliable data evidence. And then, as I said, disease registries play an increasing role, and I think this is a nice conclusion for this for the disease registries. Thank you.
Thank you. Thank you very much, Dr. Kurz, Xavier, for this nice conclusion. We have in chat, and I have one question with real-world evidence. It's a new world, a new area that is open. Do you have the same confidence about pre and post authorization power of this method for efficacy and for to exploring the safety of medicine? And a little more perhaps a philosophical question. Do you think that this new method and the power of this method are liable to modify the concept of modify some our scientific concept of causality, because we will have some facts, some real-world evidence, but don't you think that we will need the same exactly the same scientific causality to manage that for patients?
Okay, I will start with your first question, but I prefer the second one, I have to say.
Okay.
The first question is, for safety, it has been used post-authorization for a very long time. That's not an issue. For pre-authorization, so I try to explain it, this is increasingly used as a supportive evidence. There are currently the views is that it's not going to replace clinical trials for sure. It can provide supportive information to contextualize the clinical trials. In some exceptional circumstances, it may replace some clinical trials. I know of one product that over the last 2 years that has been authorized only based on real-world evidence, but this is exceptional because of the type of product and the type of disease and so on. So for efficacy or effectiveness, for effectiveness, it can be useful, but it's not going to replace efficacy. So once the efficacy of a product has been demonstrated in clinical trials, real-world evidence probably can investigate some effectiveness aspects in real life by looking at the effect of different dosage, by what we call effect modifications, the factors that may influence the efficacy, but once the efficacy has been demonstrated by clinical trials. Now, regarding the second question, it's interesting because this terminology of real-world evidence has been included and been used in the regulatory world a couple of years ago. But this is a bit broader than just observational data, but we have to keep in mind that what is important in terms of methodology, that in order to use real-world evidence, analyze real-world data, real-world evidence, we need strong method, and this is pharmaco-epidemiology and statistics. So there are no specific methods for real-world data or real-world evidence. There are applications, I would say, fields of applications of statistics and pharmacoepidemiology in terms of methods.
Thank you. One question on the chat: what is the estimated time frame for conduction of the 4 types of studies described in days, weeks, and months, considering the rapidity required, for instance, in the event of a serious adverse event?
Yeah, okay, so what is planned is that the rapid analysis could be done in a matter of days, couple of days. More complex studies now with the framework of common data models that has been explained by Edward Byrne and that will be applied in DARWIN EU. When a new issue to be investigated, probably we're estimated to, let's say, realistically, couple of months. Yeah, probably that they Anton showed study being done in 1 month. I think this is specific circumstance. I think it is probably possible, but in specific circumstances, it is probably exceptional. But if we have several studies to do at the same time, I would say a couple of months. Yeah. Okay, thank you. And the last one, tomorrow we will have a roundtable that will address the place of the patient. Could you tell us how you are going to include the patients in your works?
I think about also the benefit risk, but also to more define the needs, the patient's needs.
No, exactly. And this is why we want to include them also, because we realize that the questions that are being asked by regulators or by academic people are not the same as the patients are asking, and they may have other type of questions than the one that we have, and we have to answer them. And for example, we are rather, I would say, poor, or we are not very good at this stage now to answer questions on patient-related outcomes, quality of life, and or other outcomes of treatments than just effectiveness or safety. And there are many others, and probably we should then listen to them to be able to see how we can better assess this information.
Okay. Thank you very much. I think that there is no more question, I suppose, and so it's time now to conclude this afternoon session. And we have seen that the setup of common data model is driven by several challenges, and is not only involving adequate use of digital tools, but also skills in analyzing the data and networking abilities, and I think also ethical consideration. And Vincent?
Yeah, then we give you appointment tomorrow beginning at 9 for the next session of the conference. We have seen today that we globally are sliding from post-authorization evidence for safety, then post-authorization data for efficacy, and now we are sliding to pre-authorization with the registry as illustrated by the presentation of Xavier Kurtz, for example. So it will be the main thematics of tomorrow. And I would like to thank Marie-Caroline Pesquidou and Rasolo Dramanitra that have taken the question from the chat. And I wish you a very nice end of the day. Goodbye.
Goodbye and thanks.